right. Welcome, everybody, to Standing for Truth. My name is Donny Budinsky, and I am your host and moderator for tonight's Great Origin of Species debate. I am thrilled to have two well-studied academics here, Dr. Jerry Bergman and Dr. Chris Thompson, for this exciting debate on origins. Dr. Bergman holds creation or separate ancestry, and Dr. Thompson to evolution or common descent. And I am looking forward to an engaging, thought-provoking, and comprehensive debate on, again, the origin of species. Gentlemen, if we could, let's get acquainted, break the ice a little bit, get to know you guys a bit before getting into our opening statements. You gentlemen are no strangers to debates, certainly not your first rodeo, nor is it your first time here on the Standing for Truth debate platform. You guys are a couple of true professionals, so I'm looking forward to this. Dr. Thompson, it's good to have you back. Let's start with you. Tell us, how have you been since the last time you were here? And just tell us a little bit about yourself.
Wonderful. Yeah, thanks for having me back, Donnie. I appreciate it. I suppose I'm professional in the academic sense in that I'm an assistant professor of neuroscience at Virginia Tech. I study the mechanisms by which environmental toxicants affect brain plasticity. I've been very busy with work, doing a lot of paper writing and dealing with grant issues and that kind of thing, the typical academic stuff. And yeah, I've been good since the last time I was here, and I appreciate the opportunity to be back again. And but I don't know, I wouldn't necessarily call myself a professional debater, at least not yet. This is still very amateur hour on this end, but I've done quite a few of these kinds of things, and I do have my own YouTube channel where I do post things related to neuroscience, but also to creation evolution. Going to be doing things on fluoride as well, so fluoridation of drinking water. I've got a number of videos in the work on that, and yeah, so that's kind of what's up on this end.
Very good, Dr. Thompson. Appreciate that introduction. For anybody who wants to see more from you, if they like what they're hearing tonight, I do have a link to your YouTube channel in both the live chat and the description box where they can see more from you. Chris will have you addicted to debates in no time, and therefore you'll be a professional in no time. Again, Chris, thanks for being here. Dr. Jerry Bergman, it is great, as always, to have you here as well on Standing for Truth. And so please tell us, Jerry, how have you been since the last time you were here? And also tell us a little bit about yourself.
Well, I've been busy writing and publishing, so I'm responsible for about 2 articles every week. So I used to do a lot of reading, but now I have to spend more time writing, which is, I guess, good because I've done so much reading. I have a good backlog of ideas. But I taught at several universities: Bowling Green State University, University of Toledo, Medical College of Ohio. And my background is biology primarily, genetics. And at the medical school, I was involved in doing research on cancer. So I'm very aware of the whole mutation concern because, of course, mutations are the cause of cancer. And so I've been interested in this issue. I was an evolutionist for a while. My father was an evolutionist, very involved actually in the evolution issue. And so I came out of that background. And the more research I did, the more I began to realize that there's a lot of big problems with it. And I've spent probably 4 years looking at the problems. I find it very rewarding because I assume, well, gee, there's going to be a problem here. And as I do more and more research, I find out indeed that suspicion has been confirmed. And so it's very rewarding. I read mostly books on evolution. I read a few things on creation, but mostly evolution. And I find most evolutionists pretty honest. And if you look at the literature carefully, you can see, although they believe in evolution, it's a belief, but on the other hand, it's very clear that the evidence is problematic, at least, and often it's overwhelming against the evolutionary worldview. And it is indeed a worldview. And so I find that very rewarding. I'm retired now, so I can spend all day pretty much reading and writing, which is what I do, and speaking.
and now doing debates, Jerry. So I appreciate that. I appreciate you both being willing to engage in these types of events. I think it's important that we come together, those of differing positions, on the topic of origins and have a good, engaging exchange on them. And so, Dr. Jerry Bergman, for those that like what they're hearing from you, in a similar way to Dr. Thompson, we have his YouTube channel linked in the description box. I have links to your articles and where people can find more from you. So again, gentlemen, thank you for the introductions. I am excited for tonight. This is the Great Origin of Species debate, and I will go over the format briefly before we get into the first opening statement. And so we have opening statements of 12 minutes each. Dr. Chris Thompson will be kicking us off with that. Then we'll be having a round of rebuttals. That'll be 8 minutes each for our debaters tonight to respond to the points and arguments brought up in the opening statements. Then we'll jump into open discussion. Now, what we have for the open discussion is 40 minutes. I've got it broken up into 20 minutes each, where both debaters, Dr. Thompson, Dr. Bergman, get 20 minutes each to lead the way, picking topics, asking questions. It doesn't have to be overly strict in terms of a cross exam, but just kind of guiding along the discussion. Then we'll have audience questions. So this is where we get you guys involved in the audience. Please, if you got a question, a challenge, perhaps you hear something that our debaters say tonight that you disagree with, send in an objection, send in a comment. Just make sure that there's a question mark at the end of it. Make sure your questions are professional, of course, and we'll have some fun engaging those. And then we'll wrap up the night with closing statements, 5 minutes each to wrap up our thoughts, wrap up our points for tonight's debate. With that, Dr. Chris Thompson, I can get your slides up when you're ready. And you've got 12 minutes. The floor is yours.
Excellent. Well, again, thanks for having me here. So yeah, tonight we're talking about On the Origin of Species. Of course, this comes from the title of Charles Darwin's famous book, where he laid out the principles of natural selection. Now, of course, yeah, as you pointed out, I'm a proponent of evolution. I don't consider myself to be an evolutionist, but we believe in common descent with modification via mechanisms of evolution, including natural selection. So we're going to be talking about phylogenetic trees and nested hierarchies, right? That is a big part of what the debate is over. And so this is a very basic rudimentary phylogenetic tree. So, and we can do these phylogenetic trees based on morphological features, but also things like molecular and genetic dating, which is going to be even a more robust way of creating these nested hierarchies. So in the nested hierarchy, it's also going to be based on when was the last common ancestor, right? And so some of this, right, all the animals here are vertebrates, but one of these animals doesn't have a bony skeleton; they have a cartilaginous skeleton, so they are an outgroup. And then if we look at the rest of the groups of these animals here, we see the ray fin fish. They're the ones that don't have limbs, so therefore they're an outgroup for the rest of these groups. So we're moving up the phylogenetic tree. So there would be a more recent common ancestor for the tetrapods. The outgroup there is the amphibians, because they don't have an amniote. And then within the amniotes, we have the primates and the rodents, which are the mammals. They have hair, they provide milk. And then there are the reptiles and the birds and the dinosaurs, where they are the diapsids, right? They have two postorbital fenestrae. So based on just morphological features, the more recent the common ancestor is, the more in common you're going to have that's going to be similar. That's going to be the basic principles by which nested hierarchies work. Now, the thing is, we're going to talk about natural selection as well, right? That's going to be one of the things about the origin of species. So natural selection is that variation exists within a population. Much of that variation is heritable, right? These are the two basic facts that Charles Darwin saw that has been known for centuries. And that variation is sometimes better suited to survive, allows those animals to survive and reproduce than others, depending upon the environment, right? So some variants are just going to confer, say, like a camouflage benefit or some sort of metabolic benefit that allows that animal to survive in a particular environment and then to survive and to reproduce. What that means is that over generations, You're going to have beneficial traits that become more common or even fixed within the population, right? So these variants are then become fixed within the population. And so if you've started with a stock population and if the populations diverged over time, those two populations may be facing very different environments. So even though there was an original species, then they split into a different population. They will then, over a long enough period of time, they will become so different that they can no longer reproduce together once they come back together, right? And that is the basic. Then you have speciation that has occurred. So I've got a little brief video here. There's no audio here. I'm just going to play the video. This is taken from the YouTube channel Stated Clearly, which is a fantastic YouTube channel. I highly encourage everyone watching to go check out this YouTube channel and to subscribe because it's filled with excellent information and really great animation. So basically what you have here is a bunch of different variants within a population of some sort of cricket. And it's going to be facing some sort of environmental challenge, such as predation. Some variants are going to be better suited for the environment than others. But then there's going to be, of course, variation that occurs through multiple generations because there's mutation that occurs that allows for variation, right? That is something that occurs. So it's these mechanisms by which natural selection occurs, where you can have an original stock population that then changes over time through generations. I think even both young Earth creationists and evolution proponents believe in these things. Now, I don't want to say natural selection is the only mechanism of evolutionary change. There's also genetic drift, migration, also mutation. Mutation is an important thing. I know Jerry's going to be talking about that. There are mutations that can be beneficial. Most mutations are neutral. They really have no effect. Of course, then some of them can be deleterious and cause cancer, just as Jerry already stated, and I'm sure he will be talking about. But there are beneficial mutations. We can see these examples from artificial selection experiments. So you can go into the lab and try to develop molecular tools, not through like designing features or like trying to create specific mutations, but instead just inducing a bunch of different mutations in the construct that you're trying to mess with, and then look to see which one actually confers the benefit for what kind of tool you're trying to develop. Then you take that and then do it another round of mutation and another round of mutation and selection, mutation, selection, mutation, selection. Through that process, you can create whole new genetic tools. This kind of technique is used all the time in the lab. We can also see evidence of beneficial mutations that have occurred in the recent past by comparing closely related species that both young Earth creationists and evolution proponents would agree are actually related to each other, such as the Maine wolf and the bush dog, which, from my last debate, you might think that we're going to spend a lot of time talking about them, and we could. But one thing I do want to also emphasize is that, comparing our model on the evolution proponent side to what the creationists believe, there's actually a lot that we agree on, and that's what I hope that we can talk about tonight. But to just set the stage for what creationists believe, they believe that all animals were created in the Garden of Eden around 6,500 years ago, and that, or I guess on Earth anyway, and then from then there was then variation that occurred, right? So some animals ended up varying to some degree, but unfortunately there was this bottleneck that happened around 4,500 years ago when God demanded that Noah takes one male and one female of each kind of animal on the ark, and that the rest of all the other animals were killed in the global flood. So that meant that within each kind, whether you're talking about the rodent kind, or you're talking about frog kind or elephant kind, you were reduced to just a single male and a single female. So then after the flood, all these animals were released from the ark, and all of the species variation that we see would have had to have evolved via natural selection and other evolutionary mechanisms within the last 4,500 years. So they also believe in phylogenetic trees, right? So within the rodent kind, there would be this phylogenetic tree as well. So this is the thing, we actually agree on a lot. And that's what I hope that we can get into today, because I want to talk about what is the species mechanism that occurred within the last 4,500 years from the Earth creationist model. They also, of course, just worth pointing out that they believe that dinosaurs were on the ark, but they didn't survive for whatever reason. All right. So you might think that I'm here to talk about carnivores, since that's what I talked about last time. We want to get into it. I'm happy to, but in fact, I want to talk about rodents. So rodents, this is the rodent phylogenetic tree. I realize that this is what evolution proponents believe, that creationists don't believe that all these animals are related to each other. But within the rodents, that we have things like beavers, squirrels, chipmunks, but also within the myomorphs, which is this branch here, we've got the mouse-like rodents, which is a quarter of all mammalian species. This includes mice, rats, gerbils, hamsters, lemmings, and voles. So let's zoom in a little bit. Okay, so we're zooming in on that box. And within the myomorphs, we have the superfamily called the Meroidea. This includes the mice, rats, voles, and hamsters. It doesn't include the birch mice or the jumping mice or the jerboas. They're found on all continents except Antarctica, and there are 6 families of muroids. And the reason why I point out the 6 families is because generally young Earth creationists believe that the families are where we have the different kinds. We're going to talk a little bit about that. So the important point here is that within each kind or within each family, evolution proponents and Earth creationists believe in those phylogenetic trees, that they are all related to each other. We just disagree about the root of the tree. So this is a phylogenetic tree of the muroids, okay? So these 6 different families right here, right, are the different muroids, okay? So generally creationists would believe that these are 6 separate kinds, but within evolution proponents, we believe that they are related to each other. It comes from this excellent paper where they looked at 900 different species of muroids and created a phylogenetic tree based on their genetic similarity. So let's walk through them. So the first one is the Cricketidae, which are the true hamsters, the voles, the lemmings, and the muskrats. It includes nearly 900 different species within this one family or kind, right? So within the last 4,500 years, we'd have to have 900 different species events, speciation events. So that includes things like the pygmy mouse, which weighs just 7 grams, and things like the muskrat, which is over 1 kilogram. Then we have the Muridae, which includes things like the house mouse, the Norwegian brown rat, but also this crazy thing known as the Maine rat, also nearly 900 different species within this one family. Within the Nesomyidae, we've got things like the climbing mouse, the bastard big-footed mouse, and the northwestern fat mouse, okay? Again, I guess a separate kind from the creationist perspective. Within the Calamicinae, there's only 10 different species of these guys. These are known as the mouse-like hamsters. They're found in parts of the Middle East. They look a lot like hamsters, but they're actually not, and they're genetically different in a number of ways. Then we have this crazy group of the muroids, which are the fossorial rodents. Okay, this is an excellent example of convergent evolution, where we have these burrowing rodents, such as the giant blind mole rat, the bamboo rat, and the zoochores. Also this thing, the big-headed African mole rat. These are just a couple of examples of these 40 different species of burrowing rodents. And then we've got 3 different species within this family. I'm not even going to try to pronounce it. These are the pygmy mice. It's tiny because there's only 3 different species, and tiny because they're known as dormice. So very cute. So the thing is, as I said, like there's a lot that we agree on, and I want to focus on what we agree on. There are these 6 families, and creationists believe in these lines that these different groups are related to each other in general. I hope, and that's what I hope to find out from Jerry, if he agrees with that as well. Like the way Answers in Genesis also sort of states what the different kinds are. But they don't agree with these lines that, for instance, the Cricketidae and the Muridae are connected to each other in that they share a common ancestry, despite the fact that things like true hamsters and voles and muskrats and lemmings and New World rats and mice, they're somehow different. They're a different kind than the many species of the Old World mice and the rats and the gerbils. And I'd like to know why that's the case. Why do they believe that they're different from each other? Why they are different kinds? And how can we decide whether animals are to be clustered together within a kind or to be separated from outside of a kind? Because that's critical to answering the question on the origin of species. And so that I will end. And I've ended my show. I'm not too sure if I was around 12 minutes. Hopefully I was.
It's as if you've done this before, Dr. Thompson. You were basically right there at 12 minutes. So I appreciate the opening statement and also the work that you put into that presentation and visuals. Sure. To the audience, I appreciate the interaction so far. Yes, I am all caught up on your questions. So again, if you have a question or a challenge or an objection, send those in and I'll do my best to save them for the audience Q&A period. Dr. Jerry Bergman, we are now going to get your slides up, which I see them here. So feel free to get yourself set up. and I will reset the timer. You have 12 minutes as well. The floor is yours starting now, Jerry. Go ahead.
Can you see my slides okay?
Yes, they look good.
Good. Okay. My thesis is evolution's true, but it's going the wrong way. And I'm defining evolution here from molecules to man. And by the way, that is the title of a textbook that is used in biology classes. And so that's really what I'm concerned about, molecules to man. I'm not so concerned about the variation in animals as brought out quite well. There's an enormous amount of variation. And as I do more research in biology, I realize, well, horses, there are some horses about the size of a small dog. Others are about the size of a dinosaur. An enormous amount of variation, which is, I acknowledge. Okay, the primary basis of macroevolution is mutations. And to produce variation that can be selected, you have to change, and that changes the protein. And also have to change the regulation of the proteins and the sequences. And Ernst Mayr said ultimately all variation is due to mutation. Ernst Mayr, of course, lived to be 99. He wrote, I understand, 10 books in his last decade of life. So he was quite the author. Theodosis Dobzhansky, mutation is the only source of the raw materials and hence of evolution. And research done a few years ago was discovered that you can speed up evolution. And that was done by Hermann Muller, who is an interesting guy. And he basically put insects under flies and other insects, fruit flies primarily, under an X-ray machine and got a huge amount of variation. And this was exciting to the evolutionists because they said that this discovery actually speeds up evolution. And in his Nobel lecture, he basically said that indeed, it's here somewhere, his Nobel lecture, thus the Darwinian theory becomes implemented. And this is an area I have an interest in because I found evolution has made so many predictions and it's embarrassing. So many of them have proved wrong. And this is one good example. So now we know that, of course, to spend time in an X-ray machine, CT is problematic. So you got to get them in there, get the CT done and get them out because we know that X radiation causes mutations, which are a problem. Now, there are other sources of variation, but we're not concerned about that. We're looking at new genes, not moving, crossing over, transposition, and so on. Richard Dawkins, he basically gave us an example of how evolution works. So what you're trying to get, a goal is me thinks it's like a weasel. That's what you're trying to get. You type in a bunch of letters at random. And each generation, the letters are changed. And this is a computer program which is set up. The letters that fit me thinks it's like a weasel are saved. And as a result, after 44 changes, we've got the result. And this is the illustration he uses to prove evolution. While there are many problems with this, which I won't go into, but a few I will mention. One is it assumes mutations are random, and that is not at all true. In fact, we get, when we look at the mutational set, we get a lot of patterns. And the random mutations produce certain amino acids, but ones they produce are more likely than others. And if you know something about genetics, this is pretty straightforward. You have 6 ways of producing serine, the amino acid. So randomly, now that's not, of course, in the organism, it's not random, but if you had random changes, 9.4% would be producing serine, 9.4 arginine, and only 1.6% tryptophan. So that's one thing you have to look at. Though mutations from one DNA base to the other is not equal. And here's one study which was done, found that you end up from G to T about 16%, C to T, and so C to T is much more common. And again, there are a lot of factors involved in this, but by and large, the conversion from one base to another is not equal at all. In fact, some are fairly low, So what's going to happen? You're going to end up with a lot more thymines. And here you can see cysteine, adenine. And if you did that, you found conversion to thymine is 10 times more common than conversion to adenine. So when you look at that, what's going to happen with Darwin's set? Well, you're going to end up with a lot of T's, and eventually you'll end up, and this is schematic, so you can see it's not actually what's going to happen, but basically you get more and more T's. Another factor is hotspots, which are really important in doing cancer research because hotspots are extremely common, and you can see when a gene is damaged, like P53, what you end up with is a lot of hotspots. And there you can see, now this is E. coli. We've done these with a lot of different genes, and you find that 199, that position on the genome, is a very common hotspot. And you can see most other areas, you get few or one change. They're very rare. So what that does is, we know what that does. It basically causes deterioration of the genome. In fact, the big question is, why in the world doesn't all the genomes degenerate because of this problem? Well, they do, and that's partially what causes aging. And we also know that there are ways of counteracting that, like the mutation repair system, and P53, which is very important. One study, they found 2 mutations counted for 94.4% of all mutations out of the 319 that they identified. And so given this hotspot and back mutations, we're never going to get me thinks it's like a weasel, given the example that Dawkins used. And when we can combine these together, we can see that they indeed will not give us what we're looking for. Polarity is also important, that you cannot end up with sets that are not polar or nonpolar. You have to have a combination in a certain place in order to produce a functional protein. And therefore, even if all this stuff, this randomness can be evaluated, you still have to evaluate the idea that you have to have certain polar uncharged or polar nonpolar, etc. So you have basically five different possibilities, and that has to be a part of the system in order to get it to work. Okay, are beneficial mutations common? Did a study of the literature and found there were, out of 18 million records, now this is a few years old, so out of 18 million records, there are only 186 that were declared in the journal to be beneficial. And I looked at all the 186, and I found in most cases they were not actually beneficial for the organism, they were beneficial for humans. And a good example is seedless fruit. It's not beneficial for the fruit, it is for humans. And another example is the beef, which was valuable to beef farmers, called the Belgian blue cattle. It's interesting, when I was in Europe speaking at a university there, I was at the university where this was discovered. So they were very interested and could tell me more about this discovery. But anyways, it was valuable to beef farmers, tender, low in fat, ideal. Well, there is the Belgian blue cattle. And you can see what happened is the muscle grew well beyond what it should, and they found the mutation that produced that. And by the way, in the end, this did not turn out to be very functional for a lot of reasons. But they found basically it has problems. What was the cause of this was the gene which stopped the growth of the meat was broken. So therefore, the meat grew beyond, the muscle grew beyond what it needed to, and as a result, you had all kinds of problems. You did have, though, some beneficial changes for the farmer in this case. And they found that they didn't in the long run work. There are other problems. Probably the most famous beneficial mutation was discovered in 1791 by Seth Wright. Darwin used this example for a number of his books, and that's it, the Yukon sheep, and that was taken from a textbook. You'll notice that's black and white because even though that was commonly used in textbooks for years, they found that that was not beneficial. In fact, what had happened was a mutation caused the Yukon sheep, which is in the middle there, to have short legs. And so it was actually a disease called achondroplasia. And there you can see in humans what achondroplasia looks like. Another example was, and I learned this when I did a presentation at Ohio State University, and the professor there said there's got to be some examples. I'll look for them and I'll send examples to you. And he looked and looked and had to prod him a little bit, but he said, I'd like to see some examples. He said, well, here's an example. This guy's got a 6-finger mutation. He had an extra finger, and you can see it was advantageous to climb trees. This guy was really good at climbing trees. Well, I did some research on that, and I found out that this was a nice example where it seemed to work out well, but the vast majority of these 6-finger mutations were something very different. And you can see a couple of examples here. Generally, what they have to do is lop off a number of fingers. Another example.
30 seconds left, Jerry. 30 seconds.
A child born with an extra arm. And again, these are interesting, but they're not in the long run beneficial. Kyle, the same thing. And you can see that, well, mutations, of course, is the cause of cancer. And therefore, we spend a huge amount of time studying mutations, and they are very, very rarely, I'm not saying zero, but extremely rarely beneficial. And I did mention that at one of the symposiums that I gave at the college, and I talked about beneficial mutations, and I got what they call the look, which means that, are you feeling all right? And many, many examples, the vast majority are not beneficial. Some are neutral, but they're near neutral, which means they add up in time.
5 seconds.
And they cause. And I'll give you one example, streptomycin. It's very effective in turning off the bacteria by blocking the ribosome. And if it develops a mutation, the active site has changed. And as a result, streptomycin cannot fit in the active site. And so, yes, it is a mutation. It's a beneficial mutation for the bacteria, but in the long run, it is beneficial because it causes damage to the bacteria, and there are many examples of that. So, spent much of my career studying mutations.
Jerry, that's time. That's time.
And we're done.
Okay, Dr. Bergman, thank you very much for that opening statement. I appreciate the visuals and the work you put into that presentation as well. Gentlemen, Dr. Thompson, Dr. Bergman, that concludes our roughly 12-minute opening statements for tonight's debate. We're now moving into a round of rebuttal. And so for the rebuttal, we have 8 minutes. Dr. Thompson, if you need a few extra seconds, go ahead, just to keep things as evenly timed as possible. And I'll give you both a 1-minute warning before you hit the roughly 8-minute mark, just to kind of start winding things down. Okay, Chris, whenever you're ready, the floor is yours, and I'll start the timer on your first word.
Great. Yeah. So if you could also bring up my slides. Again,
yes.
Slide I'll talk through. Great. So, and I don't know if I'll necessarily need the whole 8 minutes. We'll see. So the thing is, I do want to rebut some of what Jerry had to say. And then I hope that he'll respond to some of what I said in my intro. The thing, we do know that mutations can induce and can lead to different function that can be beneficial. For instance, and as I mentioned, like we actually use the principle of evolution via natural selection, in this case, artificial selection, to evolve new technology that can be used in the lab. So there's this whole series of receptors. I even use these receptors in my lab, in my own research. They're known as DREADs. They can be used to control neural activity. So you can take a receptor, such as a G protein coupled receptor, for instance, an acetylcholine receptor. So it's sensitive to a neurotransmitter such as acetylcholine. It'll bind to the receptor. It'll induce changes in neuronal activity. What the principle of dreads is that via the process of artificial selection, so taking the gene for the G protein coupled receptor and modifying it, just adding in random mutations, and then selecting for those variants that actually are now sensitive to the designer drug and not so sensitive to the neurotransmitter anymore, you can make a new receptor that did not exist before that can now be used to modify gene neuronal activity by just using the drug, and it's no longer sensitive to the neurotransmitter. And this has been done several times, many times. The title of this paper is called Evolving the Lock to Fit the Key to Create a Family of G protein coupled receptors potentially activated by an inert ligand. So basically, the way that this is showing like the basic activity. So with this acetylcholine in the first generation, you get activity that goes up at a very low concentration of acetylcholine, but not so high for CNO or clozapine. So CNO is the designer drug that they're trying to do here. So then they did a whole bunch of different mutants. So they go through and they do a bunch of mutants doing this in yeast. So they introduce the gene into yeast. They have a bunch of different mutants. They find one variant that happens to have some sensitivity to clozapine and then less sensitivity to acetylcholine. And then over time, the blue line is the clozapine and CNO is the designer drug. Over time, basically what you can see by generation 118 of doing this mutation and then selection, you can get now a receptor that is no longer very sensitive to acetylcholine, but is now sensitive to CNO and clozapine. So you can evolve new function through mutation and selection. It's this principle that can be used, and this can be done in a matter of weeks in the lab. And so using this technique, you can now put it into the brain using a lentiviral transfection vector. So like I said, this has been done in a number of different kinds. So we have CNO dreads that can actually inhibit neural activity or excite neural activity. You put it into certain parts of the brain, and now all you have to do is just give the animal the drug. Either just let it ingest the drug or you inject the animal with the drug. It will act on those neurons that have taken up this designed receptor, this artificially selected receptor, and it'll affect neural activity, either increasing it or decreasing it. And then you can put them into certain kinds of tasks, such as putting them into social defeat or social avoidance tasks. And then by introducing the drug, you can actually change the activity of the animal depending upon whether you introduce CNO or not. So this is just an example of how this can work. Now, I also want to get to, I talked about carnivores in my last debate. I want to get back to that because I do want to talk about the Maine wolf and the bush dog, because we do need to talk about species, right? We're talking about on the origin of species. And I'd like to know if Jerry thinks that like the dogs are considered a single kind or not, or are there multiple kinds within the dogs? The reason why this is important is because we need to know, like, how did speciation occur within kinds? I think that's really critical to what we're discussing here. And I didn't hear anything that he had used to address that. He just talked about mutation. So let's talk a little bit about this, right? So we got the Maine wolf and the bush dog. You can see that, so that in this phylogenetic tree of all the canids, actually this doesn't include all of them. It's all the ones that were included within this study shown here. But it's most of the canids. We can see that they all fall within a single phylogenetic tree. So presumably young Earth creationists would say that this is, they all came off the ark, and then we had a bunch of speciation that occurred during from that single male and single female pair that was on the ark. So we got the Maine wolf and the bush dog. You can see that these are sister species, but they're radically different to each other. Okay, and here's another paper where they looked in depth at the South American canids, showing that they are much more closely related to each other than they are to any others, including the Maine wolf and the bush dog being sister species to each other. And the thing that's crazy about that is that these two animals are more similar to each other than they are to any other canid on the face of the planet, yet they look incredibly different from each other. They're more closely related to each other than any other canid, but this is what they look like. One is short and stubby and has webbed feet and lives in the marshlands, and the other lives in grasslands and eats wolf apples. Okay, So based on the genetic evidence, it indicates that they had a common ancestor around 3 million years ago. This is the bush dog. Again, I said it has webbed feet. And here's the Maine wolf. And this is the apple that it eats, right? So they have very different diets. They have very different environments they live in, but they are more genetically similar to each other than they are to any other animal. And so in this study, they fully sequenced all the genomes of these animals, and they were able to identify what are called private alleles. And they found more than 100 different private alleles in bush dogs and Maine wolves. And a significant portion of them, nearly 10% of them, are related to just things like limb length. So one thing that seems pretty clear is that if they are shared a common ancestor, then because they have private alleles, these have to be new mutations. They cannot be mutations that were found in the original pair that came off of the ark. It's impossible. These are new mutations. They have to be new mutations. Okay, I'm going to say that again. These are new mutations. They were not on the original ark. Some of these mutations are in genes that are related to limb length. So several of them are shown here that we know affect limb length. And so within this paper, they hypothesized that this could lead to the differences on why the limbs are so long in these animals and so short in these animals. These are new mutations that obviously conferred them a benefit that must have evolved that allowed them to adapt to the environments that they live in. Other things, for instance, the Maine wolf, it underwent evolution related to butyrate metabolism, which is critical for digesting fruit fiber, right?
1 minute
The
left.
number of mutations that they have, right? It's shown right here. And within the pathway for butyrate metabolism, that these enzymes have been evolved and have undergone clear selection within the Maine wolf. And since it gets nearly 50% of its calories from just eating fruit, that makes a lot of sense. So clearly that these are beneficial mutations that must have occurred in these animals. And again, they're more similar to each other than they are to any other canid. So either they are separately created kinds, right? Which I think is ludicrous because they're more similar to each other than say chimpanzees are to humans. Okay. So you'd have to explain why they're so similar to each other, but they also have unique mutations that are specific to each species that you cannot find in any other canid. So they could not have existed in the original stock ancestor that came off of the ark 4,500 years ago. So these are new mutations. That's what private allele means. So Jerry, you have to be able to explain this. How do private alleles come about in these canids when they obviously must have shared a common ancestor?
Dr. Thompson, thank you very much for that rebuttal. We will now hand it to Dr. Bergman. I will reset the timer, and you also have 8 minutes for a response. The floor is yours. Go ahead.
Okay, I have no problem with these minor changes. These minor, there has to be a system which allows animals to adapt because we have so many animals and so much adaptation is necessary. The horse, I just recently did a paper on horses, and it's amazing the adaptation the horses have in order to live in very cold areas, likewise very hot areas, likewise very areas that are very difficult to survive in, that have low levels of water, high levels of water, etc., very moist, and so on. So these levels of adaptation I have no problem with. What I'm saying is, how does a neurotransmitter evolve from nothing? And the air, I've looked at it, for example, is the evolution of organs in humans. And the spleen, how does that evolve? Well, there's no evidence that it evolved. An animal either has a spleen or it doesn't. Now, sometimes animals have systems that take care of the same things that spleen does. But on the other hand, it's clear from the literature, livers, the lung, the thyroid, there's so many structures, the brain, there's so many structures which there's no evidence for evolution. Either an animal has a heart or it doesn't. And a heart, by the way, I thought would be the easiest to use to support evolution, but I found out it's not, because there are systems that function as a 1-chamber heart, a 2-chamber heart, a 3-chamber heart, a 4-chamber heart. So I thought, well, gee, evolutionists probably have a good argument to show from 1 chamber to 4 chambers, and it turns out they themselves admit they didn't, that indeed the designs are very different, and they can't imagine how a zero chambered heart could evolve into a 4-chambered heart. And so they admit that it's not there. And so each organ I've looked at, and I've looked at quite a few, the kidneys, the liver, of course, the lungs, the thyroid, etc. And I found indeed the literature is very clear. We have no idea how these organs could have evolved. I think one of the most interesting examples is the tongue. The tongue, it's small structure. It's not very large. On the other hand, without a tongue, pretty hard to not only talk, but it's pretty hard to do a lot of things like eating. Fish don't have a tongue. They don't need one. They open their mouth up and swim forward, and they take in the nutrients in the water. But most, I think all terrestrial animals need a tongue to survive. Without the tongue, they are going to have a problem if they can survive. And so from my research on the tongue, indeed, they admit we have no idea how the tongue could have evolved, when it evolved, and indeed the steps of the tongue evolution were totally baffled. And so this is quite clear in the literature. And so we're not looking at minor changes. I'm looking at the evolution of various structures like the tongue. Like the eye, of course, a better example. In fact, often they are so different that instead of stating one eye evolved into the other eye, the hypothesis is the eye evolved separately, 30-some or 32 or 34, depends on the literature that you consult, but they basically claim it evolved many different times. Instead of one, in this case, eye evolving into the other type of eye. The inverted, for example, evolving into a verted eye design. So they are very, very different in design. So this is what I'm concerned about. It's true, it's a good question to point out how indeed all these minor changes occur, but I believe that there is inbuilt adaptability which allows these changes to occur. And I think there's systems that allow the genetic changes to occur or cause them to occur, which are part of the ability an animal has to adapt to different environments. Get some water here for I still must be upset trying to get on. I wish computers would evolve a little bit, so we have fewer problems with some of the things that we have. But at any rate, it's fairly clear from the literature I've looked at. Also been interested in the evolution of different animal groups, and I found all the animal groups I've looked at, clearly there's no evidence for the evolution. Like, for example, insects, where do they evolve from? Birds, where do they evolve from? There are theories, there are ideas put forward, but all of these are very, very, very problematic. And this is something I think we need to consider when we look at evolution. So a lot of the examples that was given here, I have no problem with. But the problem is, how do we get from no tongue to a functional tongue? How do we get from non-kidney to a functional kidney in vertebrates? And so that's the information I looked up and spent a lot of time researching. And the evidence is very clear from the literature. Indeed, they have no idea. It's a mystery. We have to figure it out. Most of the literature, it's clear they believe the kidneys somehow evolved, but they state, well, we'll have to do more research. We haven't figured it out yet, but we will. And so I find in looking at the literature, it's very, very supportive of my worldview, because indeed, most admit that how did the tongue evolve? We have no idea. It's a mystery, but it did. We know it did, but we just have to look at more evidence. And one of the main reasons they use for the problem of the tongue evolving is it's soft tissue. It doesn't preserve very well in the fossil record. So there are reasons they give which try to explain it away, but after a while, it's you can see that it's a problem in trying to explain it away by saying, well, it's soft tissue and therefore it doesn't preserve well in the fossil record. But anyway, that's my response. And again, I have no problem with his examples that he gave, and I think that's clear. I have a question as to how they develop, but I feel like epigenetics has shown that indeed an organism can respond to the environment and bring about changes, genetic changes, which allows them to adapt to different environments. So I think epigenetics is a built-in system, complex, incredible system, I think. And this, I think, is a good example of how indeed organisms are built, they're structured to respond effectively to the environment. And so that's my response.
Thank you very much, Dr. Jerry Bergman, for that rebuttal, roughly 8 minutes. Gentlemen, that concludes our opening statements and our rebuttals for tonight's great origin of species debate. Now we're moving into everybody's favorite part of these exchanges, the open discussion. Now, how we have it structured is 40 minutes in total, but we're going to break it up into two 20-minute sections in order to give you both the opportunity to ask the questions you want to ask and bring up the topics you feel are most relevant to tonight's topic. Dr. Thompson, let's give you the first 20 minutes to lead the way. And so whenever you're ready, just make sure to click unmute, and you've got 20 minutes. Go ahead.
Okay, I'm ready. Donny, if you don't mind bringing up slides. I'm a professor, so I can't talk without having slides in front of me.
No worries. I don't mind visuals.
Okay, great. Yeah, I figure it's good for the audience. I mean, especially when you're talking about a complex topic like this.
Yes.
So, all right. Jerry, hopefully you can see this as well. So, I'm glad that we can agree on a fair amount and that you don't have much problem with what we discussed, especially since, or what I had to say, especially since the topic of the debate is on the origin of species, and we're supposed to be talking about speciation. I'm happy to come back and talk with you about, say, the evolution of complex organ systems, things like the nervous system. That's something that I know and can appreciate. There's a lot of ideas about the evolution of the nervous system. But of course, basically all animals except for sponges have some sort of nervous system. So it's something that, like you pointed out, goes back a very long time. So when we're talking about the origin of species, though, like we're talking about speciation and things happening within a tight time scale, either on the evolution side, on the order of millions of years, which I would consider to be a tight time scale on the order of like 1 to 3 million years, but especially on the creationist side, which is like 4.5 thousand years ago. And so I want to talk about like speciation that can occur within kinds. Do you think that speciation can occur within kinds? Do you believe that there was speciation that occurred within kinds for the pairs of animals that came off of the ark?
Well, you have to define speciation, I think, and there are different definitions. One is two animals that can reproduce, they're the same species. If they can't reproduce, they're not part of the same species. So I have a whole book done by a taxonomist, and she does a really good job of explaining that it's defining speciation is not easy. And
No, it's not.
She feels, I mean, you try, and of course you can, but I mean, obviously humans are different species than dogs. So you can draw some fairly good lines, but on the other hand, there's so many examples where two animals were seen as the same species, and then somebody else came along and did more research and found, well, they're really not.
Sure, right.
So,
and molecular evidence, like, because a lot of that's based on morphology, but then we look at the molecular evidence and we find that actually, wow, there's actually quite a bit of difference between these animals. And then if you try to put them together, you see they don't reproduce together. Even though they look morphologically similar to each other, it turns out they're actually quite genetically disparate. So that they shouldn't be considered the same species. Yeah, that happens quite a bit. So that means that speciation has certainly occurred. What, like, for instance, how many kinds do you think were on the ark? Do you have any idea? And where do you put the line at kinds? Like,
I haven't looked at that.
Okay,
question.
so like the crocodile and the Muridae right here. So like the true hamsters, the voles, the lemmings, the muskrats, and the New World rats and mice. Answers in Genesis considers this to be a separate kind from the Muridae, which is the many species of mice, Old World mice, the Old World rats, and the gerbils. Like, do you consider these to be separate kinds?
I really haven't looked into that at all. Again, I just don't have. I know there's a lot of arguments on what group is part of this species, etc. And
not species, kind. Like, so that's different because these are families. And if I can go, I'm going to go to my next slide here. Let me click on this. So this paper looked at, like I said, 900 different species. What we're looking at here is like a summary of the basic groups. So, for instance, here's the Gerbillinae, which I assume is the the genus for the gerbils, Murinae, Dominae. These are all murids, which are part of one family, which again, Answers in Genesis considers to be the same kind. But here is the phylogenetic tree for all 900 different animals that were included within their study. So, for instance, this is the Muridae right here, and then this is the Cricketae, and then over here we've got the Nesamianae. Here's the Spolacinae, which is a separate family. Here's the Platacanthamyae, which is the one that has the three pygmy gerbils. These are all the separate families, right? So when we do the genetics, they all nest into this one hierarchy, but even within a kind, there's still a nested hierarchy. For instance, the house mouse is right over here. So as I said, this is one kind of these, in this study, they probably included around 400 or so of the 900 or so different species of Muridae. And we can zoom in and you can see here's Mus musculus showing this phylogenetic tree of all these different species. These are all different species. Some of them, especially the ones that are more closely related to each other genetically, they probably can interbreed, but you're not going to get an animal from here breed over here, which is where the rats are located. Rats cannot breed with mice. They cannot create fertile offspring, even though they're all Murinae, which is just one of the subfamily from the Muridae. So do you consider all of these to be one kind, or are they like separate kinds?
Well, I would say if they can interbreed pretty regularly, then they're probably one kind. But if they can't, then they're less likely to be one kind. But again, I look at the flexibility. We see a lot of flexibility in the living world. And the question I have is, how do we explain this flexibility? And I think this flexibility is built into the system. And there are many examples of that. The most recent one, which I can't really comment on, but somebody's doing a study of people who were slaves and were in this country for 100, 150, 200 years. They're finding that they are, now this is partially true because of intermarriage, but it's also, they're claiming that the environment has caused people, there's traits to be more Western and less African. People who now are in Africa, the traits are more African. And so they claim you can tell when somebody is a recent immigrant from Africa, or one has been brought over, say, as a slave 300 years ago. And so I'm wondering how in the world this adaptation could occur so that more and more African American in America, the longer they've been here, the lighter their complexion and the less African American traits that they have. Now, again, I think part of that's due, of course, to intermarriage, but on the other hand, it appears to me that they are respond to the environment changes in America, differences in America compared to Africa. And so that's my, but I realize we have a problem.
But they're all the same species. So we're talking about like how speciation occurs. And that was kind of my question. Right. So if you consider because, we're talking about like nearly 2,000, 3,000 different species just within these six families, right? At least 2,000 that have been described. And some of them can interbreed, but most of them cannot. You consider those to be separate kinds? So Noah had on the ark a male and a female of each of these kinds?
There are a lot of reasons, though, two animals can't interbreed, and they may not be able to interbreed for a lot of reasons which have nothing to do with their ability to do so. Artificial insemination, in the area of dogs where I'm aware of, artificial insemination, they've been able to get dogs, quote, interbreed, not naturally in the wild, but through artificial insemination. And as a result, two animals that normally rarely interbreed can be interbred by artificial insemination. So I suppose if you include... I guess
I'm just kind of wondering, like, how do you determine whether two species are part of the same kind or if they're in a different kind? Because, like what we're talking about is like whether they're related to each other. And I like we can base that on genetic evidence. Like all the animals here have an enormous amount of genetic similarity, and yet they fall into a phylogenetic tree where clearly some of them are much more similar to each other than they are to others. So there's this nested hierarchy. This is the part of the phylogenetic challenge, is that when you compare, especially if you're talking about molecular evidence, you're going to find if you have 3 different animals, 2 of them are going to be more similar, and they're from different species. 2 of them are going to be more similar to each other genetically than they are to the third. And then if you go outside of that, then you're going to find those 3 are more similar to each other than another one, right? Like that's how nested hierarchy works. And, within creationists and evolution, we both agree that animals can speciate, that you can have a group of animals that have diverged over time and under different environmental pressures have resulted in separate species that can no longer interbreed. At least that's what I thought. But I hear you say that if they can no longer interbreed, that means they are separate kinds. Is that what you're saying?
Well, partially, but again, I must admit that creationists debate this among themselves a lot. And there's a lot of disagreement among creationists on the same question that we're trying to look at. And that's why, to be honest, there's certainly 2 animals that will never interbreed, like alligators and horses. That's true. But on the other hand, there are so many cases where they can, under certain circumstances, interbreed. And therefore, I just let's just look at the basic differences and try to see whether or not, horses, crocodiles, impossible. Different kind of dogs, well, maybe not. And
so let's I get that. Most creationists will debate. I'd like to take a test with you then, right? So this is the domestic dog and the gray wolf, and they can interbreed, right? You can get hybrids between them. I assume that you will agree that they share a common ancestor, right?
Oh yeah, no problem.
Right. And we know this because there's significant genetic evidence for this, right? So this is one of the first publication of the dog genome where they clearly showed the similar, they basically assessed when the dog was domesticated from the wolf, and that there were these 4 bottlenecks that occurred within dogs, and so you got these 4 separate major groups of kinds of dogs, and that's from there, that's where we get all the variation that occurs within dogs. So you agree that at least they're from a shared common ancestor.
Oh, now, yeah.
Okay.
Also, there are people who have wolves as pets and find they, in some ways, are better pets than the so-called domesticated dog.
So you agree that these 2 animals share a common ancestor, right? They share a common ancestor, and this is a coyote. Do you think that these 3 animals shared a common ancestor?
I would say yes.
Right. They probably did, because 1, we also know that they can, wolves and coyotes can interbreed, including with dogs. So that's one indication that they may have been from the same kind. So we agree on that. And we know this also from the genetic evidence, right? Dogs and wolves share an enormous amount of similarity when we compare their genomes. And then coyotes, less so. So that means that there was a more recent common ancestor between dogs and wolves than there was between coyotes and the dogs and the wolves, right? You would agree with that. That has to be true.
Yeah.
Right? Yes. Okay. So we agree. So then let's keep going. So we got the gray wolf, the coyote. They have a common ancestor. The golden jackal. Do you think that these share a common ancestor?
I would say yes.
Probably, because they can also interbreed. And for instance, here's the phylogenetic tree. So we're building the phylogenetic tree. We're agreeing on a lot of this. So, but these are separate species, right? Even though they can kind of interbreed, we do consider them to be separate species based on the genetic evidence. So the golden jackal, the dog, the gray wolf, and the coyote. Here's the phylogenetic tree showing the similarity between them.
Okay.
So now we've got the domestic dog, the gray wolf, and the bush dog. Do you consider these to be sharing a common ancestor?
Yes.
Okay. And how about the Maine wolf and the gray wolf and the domestic dog?
Yes.
Okay. And the raccoon dog?
Well,
I know you may not know what it is,
but it probably does.
It lives in Asia. It's called a raccoon dog because it has these these black markings around its eyes. Okay, so let me go hold on. What I was going to talk about. That takes me to this. Okay, so this is the phylogenetic tree that I showed earlier with the raccoon dog, the Maine wolf, and the bush dog. So we were looking at this here, where these guys can interbreed, but then they cannot interbreed with, for instance, the Ethiopian wolf, the dhole, or the African wild dog. They cannot interbreed, but they're similar to them, right? Genetically similar to them. You said that the bush dog and the Maine wolf share a common ancestor with the dog, gray wolf, and the coyote, but they cannot interbreed with the gray wolf or the dog. We know for a fact that they cannot. In fact, they're from a very separate clade, which is known as the South American canids. And then the raccoon dog actually isn't a dog at all, and in fact, it's part of the fox family. So it's part of the what we call the foxes, which is its own separate clade. Now, phylogenetically, they all fall into one clade known as the dogs. Now, if you consider them all to be the same kind, does that mean that there was only one male and one female dog, wolf, fox-like ancestor on the ark?
Yeah, that sounds reasonable.
Okay. So that means that all of the variation that we see here, all of these different species, are you saying that that was included in that single pair, that one male and one female?
Well, that'd be a guess.
How do you explain private alleles then in these different groups?
Well, I know, again, creationists have been arguing among themselves about this for decades. And I try to look at the more distinct differences like horses and crocodiles instead of, and you're giving a lot of good examples, but there are a lot of other good examples which creationists have to deal with. And I kind of leave it to them to work out all these details. I'm concerned about the big where do wolves come from, where crocodiles come from.
But tonight we're talking about species and on the origin of species. And so the thing is, like, you got to be able to explain the evolution of novelty. How do you explain evolution and novelty in these animals? So, for instance, the Maine wolf, right? As I mentioned, it's a wolf-eating animal. And then the bush dog is an animal that is hypercarnivorous. These two animals are more similar to each other than they are to any other canid. You can see that here, right? That's what this phylogenetic tree means. They're more genetically similar to each other than they are to any other canid, yet they have a radically different morphology, radically different diets. We both agree that they share a common ancestor. What is your explanation for the radical differences in their basic phylogeny and their underlying metabolism and their underlying diet? How do you explain the origin of those things?
Epigenetics, I would say, is one good explanation for it.
But epigenetics only is a modification. It's like, so for the audience, if they don't understand, an epigenetic change is not necessarily a permanent change. It can be a very temporary change that is made, like a methylation that is added to DNA that changes whether a gene is expressed or not. I know you mentioned this earlier in your response. Epigenetics doesn't actually result in change in the genetic code. It cannot explain private alleles. Private alleles, these are genetic changes. So like the A, G, C's, and T's is different in this species, and it's not found in any other species. That's what it means to be a private allele.
Well, that's true. And
epigenetics has nothing to do with it.
It turns on and off different genes. That's true.
Right.
But I suppose when you're doing these comparisons, you have to determine which genes are turned on and off. But a lot of changes from my experience the changes are, as the genes themselves, that's true, are not changed, but which ones are activated and used are changed.
Right.
And therefore, this may show up in the sequencing.
Yeah, and the sequencing shows that there are distinct mutations in bush dogs and Maine wolves that seem to confer some sort of more radical, obviously morphological change or some change in their diet. But like, because they share a common ancestor and because these are considered private alleles, like there are mutations that you find in a bush dog that you cannot find in any of these other species. There are mutations that you can find in the Maine wolf that you cannot find in any of these other species. That means that they must have happened after this split between the common ancestor between these two. They could not have been in a common ancestor going back. That you and I both agree, right? We both agree in this phylogenetic tree. We just obviously disagree on the timeline here, which obviously is another problem that we probably should discuss. Right? Like I would say that this is 9 million years ago. You would say that this is more like 4,000 years ago. But the thing is, we both agree that these mutations would have had to have resulted after the split between the common ancestor between the Maine wolf and the bush dog, between this animal and this animal, right?
Well, my question is we know that mutations have a problem like hotspots, etc. We know there are very few mutations that can be beneficial. No, I'm not denying that these are occurring, but I'm saying there's another mechanism that is important in allowing these to occur, and they're not purely random. Some may be purely random,
Sure.
but I would say most, or many at least, are not purely random mutations, because we know what happens with random mutations. We know that it doesn't improve the genome. We know that it causes deterioration of the genome.
Right. I think, right. I certainly agree. And obviously I agree with much of what you had to say in your introduction, where there's a lot of non-randomness in mutation analysis, right? Like it's not truly random in that these things are likely to occur. Like they're obviously, as you know, there are hotspots in the genome where you can get more mutations than others. A lot of that has to do with the chromatin folding and the availability of the chromatin to receive insults that will result in greater likelihood of mutations to occur. The thing is, you're still not answering my question, which is, what is the source of the private alleles for, say, the Maine wolf that allows it to have a distinct genome from that of the bush dog or from any of these other animals? And the same for the bush dog. Like the bush dog has distinct private alleles, which means that it has genetic variation that is distinct just to bush dogs that you cannot find in any other canid.
I would say there's a mechanism there which we don't understand. And what I find all too often, and I've done a couple of papers on this, is that evolution gets in the way because we assume certain things are the result of evolution, and we're not looking for the actual cause of the changes. And
Do you have any idea what that might be?
We would not. Well, I don't know, and I don't think it's known, and I think therefore we need to look into try to find out why indeed these changes have occurred. They clearly, as you brought out, clearly have occurred. The question is why, and to label it with evolution doesn't answer the question. To rate it as mutations again doesn't answer the question. What we need to do is try to find out specifically what is causing these changes. And my supposition is, and I would say in general, scientists don't know. So I'm in the same boat they are. But in general, what they have to do is try to find out why these changes occur in this very organized way. Whenever we see something change,
it's not so organized, right? Because these are just, they appear to be just, they look like random mutations. They really do. They look like probabilistic, just sort of random mutations. There's nothing to indicate that it looks organized.
Well, that I question that, and I think we need to look into more detail as to why.
But how, okay, so when you say organized, do you mean designed?
Yeah, a system that is designed. Yeah.
But how can that possibly occur if all these animals are descended from a common ancestor? One male and one female that came off the ark.
I'm thinking of what Barbara McClintock discovered, which of course people did not accept for a while. Finally, they did when she finally was able to prove it. And of course, now she ended up with a Nobel Prize for what she discovered. But the idea that genes actually cut out of one area and move somewhere else, go to another area. And this, at first, at least they thought this was random, but of course, now we know it's not. But on the other hand, by and large, how in the world does this occur? It's almost as if somebody is removing the genes and putting them somewhere else.
Okay. The thing is, like, these genomes have all been fully sequenced, Jerry. And the thing is, like, you can compare. When I say that we're talking about mutation, we're talking about essentially SNPs, so single nucleotide polymorphisms.
Yeah.
These are just like a single nucleotide that's been changed that results in amino acid change, which means in a change in the function of, these are functional changes, right? That's why they're important. So this isn't whole genome or parts of genome snipping out and being moved via Barbara McClintock. That actually has nothing to do with what we're talking about here. Basically, like, these are single nucleotide polymorphisms. The genome is essentially the same, except that there's a single nucleotide polymorphism that is different, that allows for now a specific allele that's been fixed in bush dogs that you cannot find in any other group of canids. And then there are private alleles that are true that you can only find in Maine wolves that you cannot find in any other canid. And the only explanation that I can think of is that if it looks like a random mutation and it smells like a random mutation and acts like a random mutation, it really should be a random mutation. You're trying to tell me that it's designed, but even Barbara McClintock's work that's not evidence of design. You're just saying, because it's non-random, that doesn't mean it's designed. Help me out here, right? Because, like,
I would say there's a reason.
Actually, gentlemen, could I jump in, Jerry, before you respond? We are a little bit over on the first part of
Sorry.
this. It's been that's okay. It's been a great discussion. It's been engaging. I really appreciate this line of questioning and therefore discussion. Let's give you, Jerry, a response to anything that Chris said there. And of course, we can continue along these lines, but we'll just do it in the form of another 20 minutes where Jerry can essentially lead the way. So, Jerry, go ahead. Feel free to respond, and then we'll move into our next segment.
Okay. My response is we don't really understand polymorphisms and why they are the way they are. We need to understand more in more detail polymorphisms, because there are so many. We say, well, it's just polymorphism and has no function or no purpose. But I wonder if indeed some of these polymorphisms aren't a result in some way of design, in that indeed this allows the production of variety that we see in the natural world, which is, I would say, by and large, not random. There may be some randomness evolved, but I would say, again, we need to, and this, I guess, the difference between evolution and creation. Creation looked for design. They look for a purpose. They look for a reason. They look for a function. And scientists maybe should look at the creation worldview and say, well, maybe polymorphisms are more complex than we think. Maybe there's a reason for them. I think one good example is we know that during sexual reproduction, you have two X's in a woman, and then one X chromosome is turned off. And except not all genes are turned off. What's going on? Now we find some of those genes that are turned off are actually switched on. So how in the world is this determination made? Well, gee, this gene needs to be switched on, and it surprised everyone when indeed certain genes were switched on. It's almost like someone that is there turning certain genes off and turning certain genes on. And now we know more and more that some of these genes are damaged and they cannot be repaired. And so then the body is able to find the gene that's damaged. Either it is not turned off or it is turned off and then turned on, and then it can utilize that gene in order to make repair in a gene that has been damaged. And so it's almost like when you talk about these things, we're talking about people noticing this and responding to that in some way. And we have to pick this gene. We'll use this gene over here, and we'll replace the gene that's damaged. And of course, a good example of that, the best example, I think, is P53. You can't really talk about it without explaining that, well, P53 figures out this and determines this, and the damage is there, so to repair it can't be repaired. So therefore, the cell will be destroyed by apoptosis. So it's almost like it's a rational response. And of course, we know P53 is a gene, produces quite a complex, beautiful molecule. But how does it do all the stuff that it does? It's almost like P53 has a mind of its own. And in some ways, I think that's true with the polymorphisms. And in some ways, I think it could be true. At least it's a possibility that we need to look at. And all too often, we say, well, it evolved, random mutations, end of story. And we need, I think, to look in beyond that and say, is there a reason for this design? And over, we see in genetics especially, there is a reason for the design. And once we find out the reason, we understand why it does what it does. For example, why would one X chromosome randomly be turned off? Why would it end up as a bar body? What's happening here? And so we need to ask more questions.
Right.
And
okay, let me jump in here. I think this might be a good way to proceed. Jerry, I appreciate that response. Chris, tell me what you think, since we are now, let's say, officially in the next 20-minute segment.
Sure.
Since Jerry brought up a couple of new points on the X chromosomes and also P53, why don't we consider that the introduction of his first couple topics and allow you to respond?
Yeah, that sounds fine to me.
Okay, go ahead.
Yeah. So let's see. There are a couple of concepts there that you're bringing in that I'd like to respond to. As far as X chromosome silencing, there's a good reason for why X chromosome would be silenced. One chromosome, so the X chromosome is much larger than the Y chromosome. So males have only one X and they have a Y, and females obviously have two X's. Some genes are turned on both chromosomes, but many of them are silenced on one. And the reason why is because of gene dosing, right? Because if both are turned on in both chromosomes, that would mean females are producing twice as much protein of the various genes that are found on the X chromosome than they would be in males. So, both males and females have 22 other pairs of chromosomes that are not differences across the sexes, and therefore having both turned on or off on both chromosomes, there's no difference in gene dosing. We know what happens if you have an extra X chromosome. That actually leads to all kinds of problems. So, for instance, Klinefelter syndrome. This is a condition where a person, generally a male, is born XXY. So they have a Y chromosome, which means they have an S or Y gene, which means that results in the masculinization of the protogonad. Therefore, it becomes a testis. But because they have two X chromosomes, they still get some of that genetic dosing. And there's some estrogen sensitive genes that are on both X chromosomes, or on one X chromosome. But if you have two of them, now you're getting the more female-like dose of some of those genes. And many of those individuals with Klinefelter syndrome grow breasts. They have like breast-like tissue. They're more prone to getting breast cancer. They can actually expel milk. And then it also turns out that there's a 10 times more likelihood of individuals who are born with Klinefelter syndrome to have gender dysphoria. So, yeah, okay. So that's my answer about genetic silencing and gene dosing. That yes, it's largely silenced. We kind of know why, because if it was turned on substantially in both chromosomes, it would actually result in substantial differences between, like huge differences between males and females. That would be very problematic. As far as P53 goes, I mean, that's I don't see it as something that's designed. I think that it's just a tumor suppressor gene that as a regulator of like mutations within the genome. There's a good reason, like the genome from an evolutionary proponent's perspective is that it's undergone natural selection, that many of the genes are highly conserved, that they've been under positive selection for so long that you could imagine that, hey, this is kind of been tuned over these periods of mutation and selection, that the emergence of a protein that provides some support for resisting mutations makes sense. That's something that you could see evolving. Now, it's way outside of my field, so I'm sort of speculating here. I imagine that there are people who work on this and who would hear my explanation, and maybe they're pulling their hair out right now. And that's okay, because I'm a neuroscientist and I don't work on this kind of thing. But that's one of my speculation about it. So, but I just don't see it as design. Like, I think it violates everything that I think of as design. So when I think of designed objects, like I think of how do I know that something is designed? It's not because I do some sort of watchmaker test to see that if it's all intricately working together. I think about, how did it get designed? Like, where did it come from? Most of the things that I have in my environment that are designed, I can see that it says like made in China mostly, right? So there's evidence of a designer because of that. That it has literal fingerprints on it. That there's not a single thing that I can think of that humans design that operates the way that life does, through cells dividing and then resulting in the developmental pathways that allow for specification of some genes to be turned on and off, and like that's just, it doesn't match anything that I think of as like an object that actually is designed. And given that we can see the novelty that occurs within kinds, right? Like if we both agree that these are kinds, I call them families, you call them kinds. You can see the evolution of novelty, and that's obviously what I mean by the private alleles as well.
Except your explanation of silencing the X chromosome is clearly creations like this example, because it's clearly designed. Why is one chromosome designed to be cut off? Well, you're talking about the dosage differences, of course, and we know that. So you're given a design explanation, very good design explanation for the design of the control of dosage levels And of course, we know there are many other examples, trisomy 21 and trisomy 18, Edwards syndrome, and so on. So we have many, many examples of that. But we are explaining the design. So there is design reasons for, in essence, silencing chromosome, but we got a few exceptions. Why? We got a few parts that are turned off, silenced, but then unsilenced, which surprised everybody. Almost as if you have thought and intelligence behind the system. And what I find in studying human biology, and I taught anatomy for 25 years, clearly evidence of design behind it. And I'll give you one quick example.
Actually, wait, can I, but let's just interject, because I'd like to sort of respond to that point. So I work with Xenopus laevis. Do you happen to know what Xenopus laevis is?
Okay.
Do you know what that is?
Well, maybe refresh my memory.
Sure, it's the African clawed frog. It's okay if you don't know it, because it's an unusual animal model, but it's a widely used animal model. Xenopus laevis is the African clawed frog. It's this very weird looking frog that's fully aquatic, comes from Africa. That's why it's called the African clawed frog. There's several different species of African clawed frog. They're known as the pinnipeds. There are the thing that's weird about Xenopus laevis is that it has nearly twice as many chromosomes as one of its closest relatives, which is Xenopus tropicalis. So basically, what's very clear that must have happened with Xenopus, and I've looked at Answers in Genesis to see where they put Xenopus. Obviously, I got to care about that, right? So of course, I want to know what they think about the animal model I work with. So they put them all in the same kind. Of course, that makes sense, right? They all look the same. They all look very similar to each other. Xenopus tropicalis and Xenopus laevis can actually interbreed, and you can get an animal that is infertile, sort of like a mule, right? Like it cannot make offspring on its own, but it actually is utterly dependent upon whether the male is Xenopus laevis or the female is Xenopus laevis for it to survive. So there's a bunch of interesting genetics behind it too. But they have twice as many chromosomes, nearly twice as many, which means that there was a genome duplication event that occurred. And so there's speculation about how that's going to happen. Obviously, if they're within the same kind, I assume that you would agree with the speculation. I mean, like you could be open to the speculation that would have occurred. Is that so the idea is that there were these two species, subspecies that diverged, and then when they came together, so there was enough differences that had emerged. When they came together, there were enough of the intermediates between both species where they didn't fully segregate, and so the genome got fully duplicated. And we see that they've been fully sequenced. So there's a ton of work done with Xenopus laevis and Xenopus tropicalis because of this interesting genetic event that occurred probably around 8 million years ago, based on the phylogenetic evidence. Okay, so but according to creationists, that would have to have happened within the last 4,500 years, whatever. But we both agree that it must have happened. The interesting thing is that within Xenopus laevis, because this is a recent duplication that occurred, is that there's this bizarre silencing that happens depending upon whether you're talking about the long chromosomes or the short chromosomes. So they have L set chromosomes and S set chromosomes. They're tetraploid, so there's 4 of them for each of. They don't have pairs. They got quads. But there's L, which is 2 of them, and then there's S, which is 2 of those. So there's fascinating genetics happening there. My question for you, so with all the silencing that's happening within Xenopus laevis and not happening in Xenopus tropicalis because they have half the number of chromosomes, is that designed? Well, I mean, I guess you just found out like what the species are, so obviously you can't.
It could be. I don't know about that particular case, but
sure, of course.
I know I have 3 degrees from medical school. Now, I'm not an MD. I have a PhD and master's degrees from medical school. And 3 degrees, spend a lot of time there in classes, and never once was the word evolution used. Never once. If it was used once in any class, I would have picked up on it right away and remembered it, but it was not used. But a lot of examples of design were used, and I'll give you one quickly. For my anatomy professor, talked about the hyoid bone, and he spent 20 minutes on the hyoid bone. And as a result of his presentation, he said very clearly, whoever designed this really knew what he was doing, because this bone and what it does and how the tongue, etc., works is really well designed. And his presentation showed that. And this is true with all of my courses where we talked about anatomy, including my neuroanatomy course, we basically, he didn't mention, of course, creation or God, etc. But it was apparent from the presentations that indeed these structures were very well designed. And
Okay,
so,
why,
yeah, you're
why is the
bringing up some
hyoid
exceptions.
designed? I'm sorry to
Pardon?
interrupt. I'm just curious. Like, what's the justification for the hyoid? Like, does it say like made in Garden of Eden? I mean, like, what's the evidence that the hyoid is designed?
Well, the hyoid bone is designed in such, it's the only bone that's freestanding. It's the only bone that is connected to other structures, including the tongue. And without the hyoid bone, you would not be able to talk. You would not be able to eat. You would not be able to do a lot of things. So how did we eat, etc., until the hyoid bone was there? And so in order to properly eat without, IVs or other means, in order to properly eat, you have to have the hyoid bone that functions the way it does. And you're not going to be able to live until the hyoid bone and all the structures, the muscles, etc., around it are in place. And so therefore, you cannot live until you have a whole series of structures that are there. And that's true with every organ. Without the kidneys, we can't survive. And so therefore, the kidneys are, now again, the word design typically was not used. It was in
Right.
the case of the
So
hyoid bone.
the thing
word
about the
is not
hyoid
used,
bone,
but
Jerry, if I
it
can
was
interrupt just briefly, the hyoid bone. Okay, so fish don't have a hyoid, right?
right.
Right.
Don't have a tongue either. Don't need
Yeah.
one.
So the fish do have some sort of tongue-like protuberance, though. Yes, they do.
Well, you do have a tongue, yeah.
Yeah, they do. They do, but they don't have a hyoid. That's because we know where the hyoid actually evolved from. It's from the second gill arch inside of fishes. So we can watch this embryonically. So if you look at if you look at the origin, so if you look at embryonically, you can see the gill arches in an embryo in all vertebrates. Okay, there are gill arches in
Right.
all vertebrates. Then you can see where they end up. So the second gill arch in the terrestrial vertebrates ends up as the hyoid. This is particularly true in mammals.
I think we lost.
No, I'm back. Hello, I'm back.
Okay. Yeah, I was wondering if it was just me or Chris. Go ahead. You're good now.
The second gill arch in fishes just ends up as a part of the gill structure.
That's a problematic supposition. And
Why is that
those of us who
problematic?
have looked into this find that these are more than problematic. They're just a lot of assumptions that are made, which are simply not true.
But embryonically, those tissues end up as the second gill arch. Like they are the second gill arch. They end up in the gill structures of fishes underneath the operculum. But in mammals, they end up as the hyoid. So, but once mammals, once animals came onto land, there's no need for gills anymore.
Right.
And we know that a lot of those bones ending up being repurposed for other things.
Well, that's an assumption, repurposing. I would say they were designed to function the way they are. And again, you study anatomy, you can see that they're designed to function as they are, and therefore you can make claims about repurposing and so on. But I think all this is speculation. You're trying to find an explanation for something and say, well, they're repurposed. And that's problematic in many ways. The point is the organism has to live, and suppose it during the evolution has to live during each stage. And that's a problem evolution has always had, because during each stage, how do you go from one structure to another structure and yet survive and do well during the transition? And therefore, that a good example is you're relating is the parts of the fish that indeed there are similarities, but you line them all up and you're assuming that indeed the fish were here and these structures changed into whatever, including the hyoid bone. But
right, yeah, and you can also talk about the ear bones, right? So those are bones that aren't necessarily free floating, but they kind of are. If you consider them three as a cluster, you got the malleus, the incus, and the stapes that make up the middle ear, and the origin of those are parts of the jaw, right? Only mammals have inner ear bones.
Well, that's the claim, yeah.
Well, it's not a claim; it's true.
Well, okay. There's a lot of problems with that, and I haven't looked at that recently, but I have in the past, and there are a lot of problems with that assumption that these bones, in essence, ended up as part of the ear bones.
Right. Well, you can look at the early therapsid mammals that where there are transitional animals where those bones are getting smaller and smaller, where they
Well, Chris, I think you may have frozen up again, but I'm assuming you're going to be back any second. Okay, you're back.
All right.
You were just talking about the therapsid.
Gotcha. Yes, the therapsid mammals. Yes. So the inner ear. So there's a series of transitional fossils. I forget the name of the species. I'm happy to provide that for you if you'd like. But the inner ear bones, the middle ear bones resulted from the forebones that make up the jaw in the other terrestrial vertebrates. And but we can see in those early reptile-like mammals and the mammal-like reptiles the transition from that multi-jointed mandible to a single mandible, with the inner ear bones getting smaller, progressively smaller and smaller, to the point where they now make up the middle ear. And there are a series of transitions that follow through that. And people have done like the energetics modeling of whether these would be functional to confer some sort of movement benefit, but also some sort of auditory benefit. And it looks like that it indeed could function as doing both things.
Gentlemen, if I could jump in real quick, what I want to do is, because we just got a couple more minutes left, Jerry, let's give you a response. Take as much time as you need. And then because Jerry had the last word for the first round, Chris, we'll give you a response to Jerry's response, and then we'll leave it with you having the last word. Jerry, go ahead. What are your thoughts?
Well, it's been a while ago since I did a paper on the supposed movement of jaw bones into the ear. My main concern is the ear is not going to function until and unless it has every single part. And it'd be a problem, it'd be waste of energy, etc., until it has and there are a lot of parts that when I was in medical school, my presentation, my focus was on the ear. And so it has an awful lot of parts, and we know that all of them must function in order for the ear to work. And if minor changes are made, it's not going to work. And if it doesn't work, you don't have any hearing. And so that's a good example of irreducible complexity. Without the malleus, incus, and the stapes, you're not going to get hearing. Without all the parts of the ear, hearing cannot occur. And therefore, I know one can make up stories, and I think all too often evolutionists make up stories because they see the world through evolutionary glasses. And I used to see the world through evolutionary glasses. I admit I can see where Chris is coming from. But on the other hand, once the evolutionary edifice falls, then you begin to see the world through no glasses at all, but you see the world the way it is. And clearly, when you see the world the way it is, especially with hearing, it has to be designed to function a certain way. And therefore, it's obviously clearly not only designed, but well designed. And when I taught the ear, of course, I got to be careful. You don't want to mention the word design in medical school. I'm getting fired. But on the other hand, I can show the students indeed that all of these parts, how well they work, how complex it is, and how in order to function, you have to have a certain assembly which is proper. And of course, when I focused in medical school, you focus on what can happen and what can happen to cause loss of hearing. And of course, we did that as well. So indeed. And the problem is, I think evolutionists see the world with those glasses. They've got to take the glasses off and see the world the way it is. And until you do that, you're going to see the world in a very distorted way. And so I recognize a number of the arguments that Chris uses because I used to at one time accept them. But boy, we could go on for 12 hours and still. And of course, I don't remember. Your brain's only so big. And maybe Chris could tell me, how can I add more information to it and still retain everything? They say, and Chris could probably respond to this, but the more you know, the more you learn, the more your brain is filled full of stuff, and the harder it is to find things. And the problem is not lack of memory. The problem is finding things. The key is it's there. You just got to get it out. And when you spend most of your time in some menial activity, you have more room because you don't have a lot of stuff in there, which makes it a lot of files to make it harder to find things. But anyways, would need to prepare separately the arguments, but there are good arguments against the idea that these jaw bones end up as part of the ear bones, the malleus, the incus, and the stapes. So, but anyways,
okay, Jerry, I appreciate that response. Chris, you being a neuroscientist, perhaps you can enlighten us on how we can learn a ton and be able to recall it quickly.
Oh, no, that I can't promise anything other than that. Yes, I agree that I very much agree with Jerry that the more you learn, the harder it feels like to recall all these various facts and figures. And I sure wish that I had the secret sauce to make it possible to recall it all, because I certainly can't remember it all. That's for sure. So one, I just want to appreciate, offer my thanks to Jerry and to Donny for, this setting up this conversation. It's more a conversation, kind of a debate, but it's a conversation. Really good. Really appreciate it. And but the thing is, I like the fact that there's so much that we can agree upon. Now, clearly, there's a lot that we disagree on. But it looks like, as far as if you're going to create the phylogenetic trees where we're talking about within kinds, that there has to be common ancestors of these individuals within kinds. And so therefore, if we're talking about on the origin of species, and that's what we're talking about tonight, there's a lot that we can agree upon, at the very least, that speciation can occur, that it can occur relatively rapidly. I don't agree with the creationists that it can happen as quickly as they are suggesting, that you can get 900 different species of the muroids within just 4,000 years, or the 34 different species of the canids within just 4,000 years. The genetic evidence just doesn't support that at all. But that it can occur, and that they all descended from a single common ancestor. Again, we also seem to disagree on the mechanism. I obviously think that the evidence clearly shows that if you've got private alleles that are within a single species that you do not find in the other species, if you're talking about within the dogs, these private alleles, that means that these are unique genetic mutations that you can only find in that individuals of that species and not in any other species, that they must have come about via random mutation. Now, Jerry seemed to suggest that maybe they were designed some way. I think that would have to mean that there would have to have been some sort of miraculous event down deep in South America. Once the animals got off the ark, they made their way to South America. Then they diverged into these different species, such as the crab-eating fox, Darwin's fox, the hoary fox, the Maine wolf, and the bush dog, that as they were diverging into these different species, I think is suggesting that there must have been some pushing and pulling to cause a nucleotide change here, a nucleotide change there, that these are designed features. They could not have been front loaded, though, in the one male and one female that came off of the ark. That's impossible, because these are hundreds of private alleles. So it's just impossible for it to be front loaded information. You would have to be able to find at least these SNPs in the other species, and you can't. So
that's well put. I think that private alleles is a subject that I don't know much about, and I don't think many creationists know much about.
Oh, sure.
This is where I get ideas from to do papers. I read evolution literature, I listen to debates, etc., and that is the main stimulus I have for writing papers in the areas I've written. And so, Chris, you brought up really good point, private alleles, that I think we need to explore to find out, indeed, can we explain these by the design hypothesis? Or indeed, does it support your worldview? So far, everything I've looked at does indeed, or is explained by the design hypothesis. But on the other hand, you've raised a good question. I'm vaguely aware of this, but not very much.
Sure.
And it's something that creationists need, because so many of us are alike. We hear these things like private alleles, and we say, gee, let's look into this. So, Chris, I would urge you to write to different creation organizations and tell them about your concerns and say, look, you got a problem here, you guys. How do you explain private alleles? Explain what it is, and get our minds thinking and get our minds moved in a direction which we can, indeed, maybe we can explain it, maybe we can't. But in my past experience, everything that's been brought up, I've been able to explain. But hey, there's always a first time.
Gentlemen, good, stimulating, and thought-provoking exchange tonight in our discussion portion of tonight's debate. Excellent debate. And you both were very cordial, and as I predicted in the beginning, very professional. And so I appreciate it. Like you said there, Jerry, debates are important because it gets us out of our echo chambers, brings us together, helps us to refine our positions and discuss the topic, and possibly issues are brought up that can result in research projects, and that's good. And so we're going to be moving into our audience Q&A. But what I wanted to do here before we get into the first official question from the audience, a major point of discussion so far tonight, gentlemen, is the private alleles point. And so what I wanted to do is just ask a clarifying question just to make sure I'm on the same page as the both of you. And also hopefully that'll help the audience, especially in a technical debate like this on the origin of species. And so just to fully understand and to give the audience the necessary information to objectively engage this, the private alleles point, Chris, is basically in the debate, you're using those to identify ancestry, specifically in dogs.
No.
Oh, okay.
Not identifying ancestry in dogs. It's true that I was referring to when we were talking about ancestry in dogs. No, this was a point to bring up that genetic novelty can be found in species, individual species that are within a kind, and but it cannot be explained as having been front loaded in the male and female that came off the ark 4,500 years ago, or in my case, it would not have existed in the common ancestor of all dogs.
Right. And so you were arguing that would point to common descent in all dogs as basically being the better explanation. Okay.
No, that's actually not exactly what I'm trying to say. So I appreciate you trying to clarify this. The fact that private alleles exist means that there must be mutation that occurs within the genome that had to have happened after the split from the last common ancestor with its sister species and all other species that it is closely related to. These are unique alleles to this species alone.
Yeah, it's a new allele. It's a new
unique alleles. That's why they're called private alleles.
It's a new allele. All of a sudden,
you cannot find it in any other species. Yeah, well, at least as far as the canids go, because in this study they were only looking at canids. I don't know.
Okay, so these private alleles are basically genetic variants that are found exclusively within a single population or group and not found in others. So a specific species.
Yeah, and they can be within a population. That is true. You can have private alleles within individual populations that you can't find in other populations within a species. In this case, right, they didn't sequence that many individuals. I think they only sequenced like 6 or 8 individuals per species. I mean, it varied a little bit. But they were only able to find these private alleles in the species. And then they had to find it in more than just one individual. It had to be like in all of them, but not in any other species that they sequenced.
Okay. And so, Jerry, if I understood correctly, your explanation during the debate would be that the SNPs, single nucleotide polymorphisms, those changes that came about after the flood, those could have been built-in non-random changes to explain private alleles in different species within the same kind.
Or there must be a mechanism which allows somehow the formation of private alleles.
Okay. And Chris, you're arguing from the common descent position tonight that after various splits, descent with modification, you have these mutations coming about randomly in different lineages or groups.
Right. And since the great thing about looking at the canids is that this is a point where young Earth creationists and evolution proponents can actually agree, where they all descended at least from a common stock. Of course, you guys have an additional problem because you think that there was only one male and one female. So you have a difficult time explaining heterogeneity when you're talking about, 2 individuals that mated. And then obviously the only way you can get further progeny is that brothers and sisters have to mate. And then after that, it has to be cousins. So you have to talk about inbreeding depression that occurs with each individual, with each kind that comes off of the ark, right? Because you're only talking about one male and one female. Like generally that is usually death knell for most populations if you're down to a single male and a single female. But somehow after the ark, you got one male, one female. Inbreeding depression did not occur, even though we see this happening today in species that are challenged environmentally, that are like endangered or nearly extinct. So but the thing is, like the private alleles, if they're within only one species out of 34 species, then that means that it had to have come about
after the flood, the bottleneck.
Yes. After.
Okay.
Yes, exactly. So at the very least, like that has to have had to occur. So they could, so I hear this like argument from, this is one reason I bring up private alleles, is because many creationists, they want to argue that it's impossible to get novel evolution via natural selection and random mutation and the other mechanisms of evolution. But within created kinds, it had to have happened, especially when you talk about a group that's as varied as the canids. And even when you compare two sister species, the Maine wolf and the bush dog, I mean, out of all the canids, you'd be hard pressed to find two species that are more different from each other. One has huge long legs, this mane, and eats fruit, and the other one is short and stubby, has webbed feet, and is hypercarnivorous. Like it only eats meat. Yet these two species are more genetically similar to each other than they are to any other canid. So they had to have shared a common ancestor. Yet they're morphologically incredibly different. And one reason why is because obviously they have these private alleles, right? Some of these alleles, and I've got a slide up if you want to show what we're talking about. This is the bush dog. This is the Maine wolf. And the private alleles are above this line, right? So this is all the alleles, and they're looking at the differences here. So what are different? What are the things that make it different? This is a score for private alleles. And they highlight some of them that are related to the ones that are in red are related to limb development. The other ones are other things, genes doing other functions. Everything that's below this are not private. So there are things that you can find in both individual species or across other species. But these are unique variations, genetic variation that you can find only in this species. Only in the bush dog you can find these variants. And only in the Maine wolf can you find these variants. So they had to have come about, evolved, whatever you want to call it, either designed or evolved after the flood or after what I would say the Great American Interchange 3.5 million years ago when canids made their way into South America.
And then if I could jump in for one last clarifying point, I appreciate this. For Jerry and his position tonight on creation and separate ancestry, Jerry, you would have to argue at this point in time a means of these areas possibly being designed to change and other non-random means like mutational hotspots, epigenetics, perhaps something along those lines, Jerry?
Right. We would reject the idea that they're caused by mutations. And so therefore there must be some other design explanation for these differences. But Chris has certainly raised an interesting question, and I think needs to be looked at. I guess my experience is I've had this over and over, these questions raised, and I was able to answer all of them in favor of the design worldview. On the other hand, hey, there's always a first time.
Now, if I could ask one question on that, sure. Yeah. Jerry, if there are mutational hotspots in the genomes of biological organisms, including these dog species, could some of these private alleles come about through mutation since the flood?
And that could be, yes, it could be because of mutational hotspots. That could be.
Jerry, and then Chris, do you think mutational hotspots is that a design feature?
Well, it could be, and it could be, but from what we've looked at, no, it's not, because it generally contributes to deterioration of the genome. And so, yeah, it's generally not, but it could be.
Generally not. Okay.
It could be a design now. Or if you're a theistic evolutionist, no problem. God just, added a few genes in somewhere along the line.
Right. But you
And because
already
Chris,
agreed.
you'd be saying, if I could, Chris, you'd be saying that a private allele would come about randomly through mutation, but that private allele is not the result of degeneration.
Would I say that?
Yes.
Oh, yeah, I don't think it's degeneration. No,
Right. Okay.
I would say these, because I don't think the genome is devolving. I don't think it's degenerating. I don't buy
Okay.
genetic entropy at all. I think that that's an entirely bunk.
As in that private allele came about not through a
Right.
degenerative mutational change.
No,
but
yeah,
through a type
so
of
in
positive.
general, like especially if it's fixed in the population, that strongly suggests that it was under positive selection and conferred a benefit that allowed the individuals to survive and thrive and reproduce. There has to be, right? We have to have some kind of explanation for why bush dogs and Maine wolves are so different from each other, right? They're radically different morphologically, behaviorally, and dietarily, metabolically. Yet they are more similar genetically to each other than they are to any other canid. And what's the explanation? Like if they really are descended from a single male and a single female that came off of the ark, you have to have some explanation, especially because the other thing is that if you look at phylogenetics, and if you want to compare, like I said, they're so genetically similar to each other that they based on molecular dating methods, we would say that they're around 3 or so million years ago was the last common ancestor between these two individuals. So but like if you look at all the dogs, and in fact we can go back here. So based on phylogeny, this would be around 3.5 million years ago. That's basically what we think. Yeah, right here. This would be good. So this shows the bush dog and the Maine wolf around 3 million years ago. The root for all of the wolves and dog family is around 10 million years ago. So that's based on molecular dating methods. Okay. So if you just do the basic comparisons and look at mutational rates, and you have, there's some assumptions that are built into that mutation rates and that kind of thing, but these are all well-founded assumptions that are based in actual evidence and research to back that up. That's the pattern that we see. That's how we make these declarations that the last common ancestor would have had to have been that so long ago. So, but if you just kind of bootstrap this onto the creation model, well, then you would put this at like 4,500 years ago was the split. So instead of it being 9.5 million years ago, it's 4,500 years ago, which would mean that the split between the bush dog and the Maine wolf would have to be 1,500 years ago.
Okay,
right?
So for Jerry's, and I can't say too much as mod, but I guess in terms of clarifying questions, and basically what you're pointing out there means that Jerry and his position tonight, the creation position, would have to say if those species go back to a common arc ancestor or common arc archetype, there'd have to be a lot of morphological adaptability built in essentially to these kinds to explain these variations and also the private alleles.
Right. That's what I'm saying. Yeah.
But the private alleles cannot have been built in.
Right. Those would have to be
why
either mutations after the fact, if I'm
Exactly.
understanding Jerry correctly, or those areas in the genome are designed to change for mechanisms possibly either known or unknown. And so, okay, Chris, Jerry, I really appreciate you being willing to answer those clarifying questions. I think that helps me be on the same page as in terms of where you're both coming from, and then also hopefully helps the audience in light of a good technical discussion. And so, gentlemen, before we move into our audience questions, is there anything you guys wanted to say on that, just to feel like you've left no stone unturned and have been able to say everything you wanted to say?
These debates are useful. I wish we could have them in the schools, but the courts have made it very clear. One is not to denigrate or criticize evolution, and that because that's backdoor teaching creationism. So these debates, I think, would be very helpful for students, but the courts have made it very clear these will not occur in the public school. And so I think the law needs to be changed so that Chris and I can have these debates and we can stimulate each other. I know debates are really a good way for students to learn. And I know when I've had debates in the past in schools, usually not during the school year, of course, but we had good audiences, and it really stimulates people to think. So we need to be able to have these debates without being worried about terminated. And that's exactly what's happened in the past, the creationists. We try to present our position and we end up losing our job. I've got 5 books I've written, which basically documents you are not allowed to question evolution. If you do, could well be the end of your career.
Chris,
Totally
final
disagree
words?
with that. Yeah, I mean, I got to disagree with that completely. First of all, no, I don't think that the creationism should be taught in public schools because there just isn't.
I'm talking about the debates we're having now.
But this debate that we're having right now really is about creationism. We're not really debating science. This is more a theological debate. I mean, if you know, like, because the idea that that the vast discrepancies that I'm bringing up over and over again, it's not debated in scientific circles. Like, this is not a scientific debate. Like, scientists don't debate the origin of private alleles. Like, we know that results from mutations that occur within the genome. Like, that we're not going to try to pretend that there are angels that are tinkering with the genome. It just isn't the case. And that miraculous events to explain the origin of the Maine wolf in order and how it's different from the bush dog, just simply, that's religion. I mean, it is, and it's okay to teach that in church, but like, I don't think that that's appropriate for public schools. Now, the other thing though, that you were saying is but debating it here, like, I think that that's fine. I think that's basically, yeah. I had one more point I needed to make, but I should have written it down because my memory is failing too. So.
It's like you guys were saying earlier, over the years of studying and consuming material, there's just so much there.
It's mostly full, and I cannot jam new stuff in there. So.
Okay, well, gentlemen, let me say this, because as Jerry said, we probably could go back and forth on this for the next 12 hours. There's so much there, and it's such an interesting, intriguing topic. Debates like this really help promote, I think, critical thinking, helps us to refine our viewpoints. And so we do have, at the end of the audience Q&A section, Dr. Thompson and Dr. Bergman, we do have 5-minute concluding statements. So if there's anything else we want to address, that those closing statements are always a good opportunity to land the plane on our positions tonight. So let's save any additional thoughts for the closings and get into now some of these audience questions. We've had a good audience tonight with some excellent on-topic questions. And so why don't we start from the beginning? What we'll do in terms of format, you've both been here before for debate, so you know how we typically run things. Whoever the question's for, let's say it's for you, Chris, you get to answer first. Jerry gets to add his thoughts, possibly rebut a point or two, and then we throw it back to you, Chris, for a final word. And then we move along that way. Okay, let's have some fun. This is good. Such a good topic. Daniel Walker. Question for Dr. Chris Thompson. He says atheist. Not sure if you're an atheist, but Okay, so here we go.
Or agnostic. But
or agnostic. We'll stick with that. So can you ask the agnostic why Darwin said there should be innumerable transitions everywhere? Every geological formation, every layer of strata, but are not found. What are your thoughts on that, Chris?
Sure. So again, I'm a neuroscientist who works on brain development and neurotoxicity. This is a question about deep geology. However, I'm going to do my best to answer this question. Basically, the fossil record is very incomplete because of the nature of how the rocks form and get destroyed. We have subduction zones where rocks get sucked into the mantle and are destroyed. If you're talking, we have relatively good records of fossils going back over more recent time. And if you're talking more recent, we're talking around, say, over the Cenozoic since 65 million years ago, and then less so. We have fewer of those rocks available from the Mesozoic from 65 million years ago to around 200 some million years ago, and the Paleozoic even older than that. It's just the problem is that it's largely incomplete, and that fossilization just does not occur in every single environment either. It only happens under ideal conditions. So you're not going to find Darwin was wrong. He's not Jesus, and we don't have a religion in science. He's not our God. He's not our Jesus. He's not Mark or Matthew or Luke or John. He's a guy who actually had some really great insights in things that honestly are just sort of common sense. That the mechanism of natural selection, it's surprising to me that no one came up with it earlier than him, but he articulated it well. And but he wasn't right about everything, and that was one thing that he was wrong on. We obviously find transitional fossils. There's lots and lots of examples of transitional fossils. Happy to talk about some of the ones, even as far as canids go, since that's one thing that I kind of know a fair amount about. But we can talk about others. But yeah, you're not going to find them in every stratum.
My response would be,
Jerry, go ahead.
The three most common areas, three most common claims of fossil record are horses, whales, and humans. And I've looked in detail on all three examples, and they all failed. My book on fossil forensics covers some of this, but my book on the apes as ancestors goes into detail, showing indeed that they all fail to present a viable set of fossils from some pre-ape, pre-man ancestor to humans. They all fail. And this is more and more widely recognized. The horse was commonly used for about a century. And now if you look at the charts we can see of the fossil record of horses, what we see is not a nice progression from a small pre-horse to a modern horse. What we see is a huge amount of variety all over the place. And there's charts that show this very clearly. Same thing is true with whales, and the same thing is true with humans and horses. These claims are made, but when you look into detail, you find they just don't. They are not viable. Their guess is, well, this must be the fossil that evolved into this one, and there are problems with all of the claims that they've made. So that's. But on the other hand, we haven't talked about religion in this debate at all that I can think of. So we could debate in schools, not talk about religion, just talk about the evidence, and which we're doing. And so I can't see why that cannot be done in the schools. Let students be aware of the controversy, and they're by and large not. Chris talked about the uniformity about evolutionists about some of these ideas. Well, there is, because it's hard to get the other side in. You come up with a discovery which doesn't fit the pattern, you're going to find it's very difficult to get accepted in the literature, because they're saying, well, there's a problem with it. We're not sure why, but indeed, we're not going to publish it. So I find, and there are a number of studies that shown the amount of censorship in evolution is horrendous. And it's a major, major problem. And we can't get at what's going on unless they are open to looking at other evidence. Now, to some degree they are, but if you have the right credentials, and if you're a bona fide evolutionist and everyone recognizes you as such, then you can get in some of the contrary evidence, like Stephen Jay Gould did. He was well recognized, although criticized later on. But on the other hand, he was able, like the horses, for example, he and others were able to show that we got a problem with this set of transitions, big problem, and they shouldn't be used. Gould made it very clear this horse evolution should not be used. There's major problems with it, and this is an embarrassment for us today. So indeed, we don't have to bring in religion. We can just talk about the data and the science, and that's all.
Sure. And I'm happy to talk about the data and the science, but the science and the data clearly shows that the whales share a common ancestor, that the existent horse species share a common ancestor, and that they both share a common ancestor.
Look at my papers in these areas.
Sure. Right. I'm not talking about the transitional fossils, but if you want to talk about the molecular evidence clearly shows that's the case. But even the fossil evidence, it's the problem with fossil evidence is that you have a snapshot. And I fully grant that that's going to be problematic, because it's just a simple snapshot. You don't have all the features that you might want of what that individual looked like, but you have enough to know that this is now a unique species that does not share features, or it's missing some features of species that are extant or found in more superficial layers of the fossil record. And that is essentially the principle of transitional fossilization. It's something that we can see over and over again within the fossil record. Are there going to be issues? Certainly that's the case, right? Like as we learn more, we're going to find, things that need to be resolved. But that doesn't mean that that there was a flood and that there was Noah's Ark and that these animals came about from Noah's Ark. So, I mean, those are the parts where we did talk about religion. I mean, Noah's Ark is religious, like that. There's no data supporting that such a thing occurred. Everything that we know about biodiversity, all the data that I showed today is actually quite incompatible with the young Earth creationist model of Noah's Ark.
Thank you, Chris.
Have you read my book, Ape as Ancestors?
I'll have to check it out.
About 6,000.
We're supposed to go back and forth, right, Donny?
In logical literature.
Yeah, let's. Okay, appreciate that, gentlemen. You both got some responses there. Chris will give you the last word in terms of a response, and we'll move on to the next one. That was for you, Chris. Therefore, let's get one for you now, Jerry. One God is now here. Appreciate the support. And question for tonight's debate, $20 super chat, and it is for you, Dr. Jerry Bergman. Do you think mutations can result in new structures or just extra structures that have the same building blocks we see in, say, a cow with 5 legs instead of 4?
Yeah, I think the evidence is very clear that mutations will not, and there may be some exceptions I'm not aware of. And I've looked in literature and I've looked in every single claim, and so far I've seen one clear documented claim that shows mutations can result in new structures. It cannot. Mutations deteriorate. They cause problems. Deterioration of the genome is a major problem. It's a major problem that we deal with in the medical field because we know that mutations have accumulated, and we know that more and more diseases, there's about 6,000 diseases now that are caused by mutations. And there's concerns about, well, once you cure cancer, you won't have anything to do. No, mutation load is producing new diseases that have to be dealt with. And therefore, I don't think we'll be out of a job anytime soon. So there's no clear evidence that mutations, mistakes in the genome can produce new structures.
Thank you, Jerry. Chris, floor is yours.
So I think that I've already shown, I'm going to come back to the private alleles, that these have to be new mutations. You can't, there's no other explanation. You can try to pretend that there's some sort of design feature for this, but there isn't. And, one of those genes is called IGF-1, and it's heavily involved with limb development. And so it would make sense. Obviously, there needs to be more study to find out precisely how this gene mutation is resulting in the novel differences between the Maine wolf and the bush dog. But that's one of just many that are found in this study. The only explanation is random mutation. We could think of other ones, but from a design perspective, it doesn't make any sense whatsoever. And so it has to be the result of mutation and natural selection. And so these are novel structures that had to have evolved. Web feeding in the bush dog. The fact that the Maine wolf is able to process root material, and it has mutations, these point mutations that are part of that pathway.
Thank you, Dr. Thompson. Dr. Bergman, you get the last word for this one. Go ahead.
Yeah, you can get modification to some degree in structures, but by and large, new structures, we're talking about lungs, liver, the heart, etc., that new mutations are not able to produce these. We know that they deteriorate these. We know that a major problem is mutations. In the medical field, that's very, very clear. And also, of course, what causes aging? Mutations. We age. There's, of course, genetic controls that produces to some degree aging, but by and large, in most animals, aging simply is a result of the accumulation of mutations. And so mutations don't improve the structures, they cause deterioration of the structures, and that produces aging. And anyone who's been around for 80, 90 years knows that nothing is as good as it used to be when they were 20. And that's a result of mutations, which causes every structure, every organ, every system to some degree to deteriorate. And many people to heavily deteriorate. It's true, some people can live to be 113 with pretty minimal deterioration, but on the other hand, that's unusual. And even people who are 110 have evidence of clear deterioration. They've just managed to survive longer than most of us. So that's my response.
Okay, appreciate it, Jerry. We'll give you the last word on that one because the question was for you. Next one is for you now, Dr. Thompson. Alyosha, appreciate the support for tonight's debate. Again, gentlemen, it really has been an excellent exchange tonight. I've really enjoyed it. And so here's the question, Chris. Number of all small organic molecules is estimated to around, and there's some big numbers there for you. How is it possible in 4.5 times 10 to the power of 9 years in evolution going to randomly explore a space that large and generate 500,000 genus level life forms?
Mm-hmm.
Go ahead.
So it's okay. One, those are not all unique small organic molecules, as far as I can tell. I'm not familiar with those estimates. Obviously, that's a lot. 10 to the 200. I think that's 10 to the 200. I'd be surprised if that's the case. But what we do know is that the genome has around 30,000 genes, and that's generally true across all vertebrates. And the fact that if we're talking about just evolution within the vertebrates, and if we talk about the invertebrates, many of them have less than that. So we're not talking about this wide range of small molecules. So if you're going to talk about a single nucleotide polymorphism in one, say, protein that's resulted in a single amino acid change that now is in two different species as being two completely different genes, that's clearly not the case. These are now basically alleles of the same gene. And that's where novelty can come about. These big numbers, they can get thrown around, and I think the math here is wrong, in that there just aren't that many organic molecules as far as I know. And but that you can get a huge range of different kinds of organic molecules just from single nucleotide polymorphisms, or even other larger genetic changes that will result in novel genetic changes, but they're still the same number of genes for the most part.
Thank you, Dr. Thompson. Dr. Bergman, over to you. Go ahead.
Yeah, there are quite a few of these estimates that have been done, and by both evolutionists and creationists. And so they vary, of course, the numbers you end up with, but they again show the enormous improbability of the evolution from molecules to man coming about. And of course, there are many well-known evolutionists who have done these estimates as well. In fact, so many problems have come up by looking at these numbers that several well-known astronomers have postulated abiogenesis in other places. And so panspermia, that life evolved somewhere else and came to the Earth, because the likelihood of evolving on the Earth is so small, it must have evolved somewhere else and came here. But the problem is, of course, we don't have any evidence that any life evolved anywhere else, and they would have the same problems, life evolving here. So you have numbers of the likelihood of life evolving here, well, you find the numbers would be greater life evolving somewhere else, like on Mars, which of course they've been looking for life for the past 100 and some years. So yeah, the probability estimates are clearly a major problem for evolutionists. And I think they just tend to reject them and say, well, we evolved here, we know evolution is true, therefore these numbers must be wrong. And so they dissipate the numbers as a result of their belief structure. So they're seeing the world in a certain way, and therefore, well, these couldn't be true. because, well, we evolved, we know we did, here we are. So therefore, end of story.
Thank you, Jerry. Chris, you get the last word. Question was for you.
I'm open to the idea that, the earth was seeded with, some sort of life form a long time ago. That's possible. I'm also open to the idea that there isn't anything terribly magical about organic molecules evolving from precursors. And I'm open to both of those ideas. It clearly must have happened a long time ago, whether both of these things occurred. It's one of the problems with abiogenesis is that it would have had to have occurred a very long time ago. We have lots of models that are interesting, that provide pathways for these things to actually occur. One that I'm particularly fond of is the amyloid world hypothesis, where you can get self-replication of amyloid proteins that then could then lead to, they apparently have binding properties onto RNAs. And so instead of it being, say, RNA first, it could be protein first, then RNA, then from RNA you can get DNA. That's a hypothesis. And I think that there's a lot of viable, interesting work that's being done in the abiogenesis field. It's obviously way outside of my field, so I'm not going to comment with a whole lot of professional expert opinion on it. But I'm highly skeptical of, calculations from creationists saying like, oh, it's just basically impossible because I put together a bunch of big numbers and therefore it must be impossible. That's just an argument from incredulity. And it's okay to say that we don't know something, or that we still have ideas and there's still things that need to be done. But that doesn't necessarily mean it's magic either.
Thank you, Dr. Thompson, for the final word. Okay, it looks like we got another one from One God Is Now Here, and this time it's for you, Dr. Thompson. $20 super chat. Appreciate the support and question. And here is the question. Are you familiar with or willing to look into the trochlea of the superior oblique muscle of the human eye? Richard Dawkins refused to consider this threading of a needle by claiming evolution is a fact.
I'm willing to look into it. I mean, I'm familiar with the superior oblique muscle. I don't know the trochlea of the superior oblique muscle. I'm familiar with the trochlea of the patella, which I have problems with because I got bad knees. So basically, trochlea is like a furrow where a ligament passes through. And so the superior oblique muscle is one of the 6 muscles that pulls on the eye to change its orientation. And it must then pass through some sort of a furrow that's within the skull. And that must be what they're referring to as far as threading the needle. That's the best that I can do with that. So I'm willing to look into it. That's all I can say.
Thank you, Chris. Appreciate it. Jerry, floor is yours. Go ahead.
Well, I've done some work on the claims of evolution of the eye, and of course have made a lot of claims. And what intrigues me most is that we have so many different eyes. It's hard to imagine evolution from one type to the other type. So therefore, they postulate that the eye evolved like 26, number depends on who you consult, but 26 different times. So if it's highly unlikely to evolve once, how can we claim it evolved 20 some different times? And of course, the eye, again, is another structure which it's not going to work until it has all the parts. It's clearly irreducibly complex. And I know they look at eye spots and so on, and there are problems, though, with that, those claims that we evolved from an eye spot to a modern human inverted eye, etc. But that's a good question, I think. And of course, interestingly, I was going to debate Richard Dawkins once, and a group in Indianapolis, the Atheist Society there, arranged a debate, and Dawkins wanted my resume before the debate commenced. So I sent it to the group in Indianapolis, and they sent it to Richard Dawkins. Richard Dawkins sent it back and said he's not debating me. So I'm not sure whether I should have been disappointed or flattered or what. but I was preparing for a debate, which I thought would turn out pretty well, but of course it may not have. But anyways, he declined to debate me, and so that's my experience with Richard Dawkins.
Oh, that's
I was kind of disappointed he backed out.
Yeah, I think that that's disappointing too. I would agree with that that he would back out. Well, I mean, you bring up Dawkins because he has what I think is a very intriguing explanation for the possible evolution of the eye, and in his book The Blind Watchmaker. It's an excellent book if you are from an evolution proponent's perspective. It still has a lot of value to it, even though it's probably 30 some years ago, and I'm sure there are a bunch of things that are out of date in it. And it certainly is missing a lot of the new synthesis that's happening over the last 30 some years. But I encourage even the creationist watching to read it, because it actually provides a really great argument for the power of what natural selection can do. And I think that I've articulated tonight that it's clear that natural selection has the possibility of shaping and molding animal life so that they can adapt to their conditions, and that it doesn't need magic or fairies or some sort of designer or designers, right? Because one thing that I think that finds so frustrating about the intelligent design movement is that they just assume that there's a single designer. When we think of design, there's lots of designers, and so they should be open to the idea that there could be lots of different designers. And I think that if we're going to take the intelligent design theory seriously, from a design perspective, you have to assume that there are multiple designers, not just one designer.
Thank you, Dr. Thompson, for that final word. Okay, we got two more questions, one for Jerry, one for Chris. And so this question comes in. Appreciate the question for tonight's debate. Ask Jerry to explain the hyperspeciation after the arc. In other words, Jerry, how from the separate ancestry model would we get all the different variations of the kinds off of the arc, and that not be considered a degree of evolution or hyperevolution, as some would say?
Well, again, I think for the arc, when you get into this, you're getting somewhat out of science, but not totally. But on the other hand, I think the animals that were brought to the ark had a set of genes which allowed for hyperspeciation after they got out. And creationists don't talk about that a lot, but they do talk a lot about Adam and Eve. How do you get from Adam and Eve, from two people, all the variation you see in the world today? Well, geneticists that are creationists have done good work on this, showing indeed that it's not a major problem to get the variation we see today from two people. So you have basically 4 alleles, and from these 4 alleles, you can get combinations, etc., that produce all the skin colors, the hair color, all the variety that we see in humans today. And this has done a pretty good job. So my assumption is, if this could be done with humans, as they've done quite a good job, and by the way, it agrees with the stuff in my genetics textbooks when I taught genetics, I think you could probably do the same with many of the animals in the ark, not just humans, but of course, the other animals. So I think the same thing is true, that the animals in the ark had a high level of genetic variety to produce hyperspeciation when they got out of the ark.
Thank you very much, Dr. Bergman. Dr. Thompson, the floor is yours. Go ahead.
There are a lot of problems with what you just said. If you're talking about a single male and a single female as a founder for a kind off of the ark, say if we're talking about the muroids, you now have 900 different species that must have speciated within the last 4,500 years from a single male and a single female. They do not have enough genetic variation to account for all of that. You cannot front load enough genetic variation into one male and one female to account for all of the variation that we can find within these kinds that we talked about. For instance, a mouse and a rat are much more different from each other genetically than a human is to a chimpanzee. And a human and a chimpanzee, if you consider the genetic variation between humans and chimpanzees, that's a lot different than just the genetic variation within humans altogether. So if you're going to allow for the kind of genetic variation that you can find within these 900 different species, or if you look at the canids, like they're also much more widely different from each other than say humans are to chimpanzees. Let alone just the genetic variation that you can find within just human beings alone, you're going to have to come up with something else. Like it's not going to be on modeling of color variation within humans or like SNPs within humans to be able to account for the vast genetic variability that we find within the other kinds. And so this question is an excellent question. Unfortunately, Jerry doesn't have a very good explanation for it.
Thank you, Chris. Jerry, you get the last word because the question was for you. Go ahead.
Yeah, I would say they've done a really good job with humans in explaining this. And I know that people in Ancestry and Genesis have worked on this. And so far, I'm aware that they've done a pretty good job. I'll leave it others to evaluate whether or not they did a good job. But on the other hand, I think they've done a good job showing indeed how many species had to be on the ark in order to produce the variety we see today. And again, there are several books, which, Chris, I encourage you to look at, and you can at least evaluate. Again, my interest is not in this area. But Chris, I encourage you to look at the results that they have. I have looked at humans, and I think it's very convincing with the work they've done with humans, showing the variety we get today. And it's true, you don't have quite the same task in doing all the animals, but on the other hand, I think they've done a good job. And maybe that's one area we need to work on more.
Thank you very much, Jerry, for that final word. And we are down to the final question that we will get to here tonight in another excellent audience Q&A. Gentlemen, I always enjoy these Q&A periods because it allows us to expand on our thoughts and to make the debate that much more comprehensive. Alyosha, $10 super chat. Appreciate the support. And question for tonight's debate, specifically for you, Dr. Thompson. How could even speciation through random mutation be viable when molecular shape space is on the order of 10 to the power of 60 plus?
And if I recall, Alyosha also asked the question about the all small organic molecules being on the order of around 10 to the 200. Okay. So I would like to reply to that because I believe that she shared, or she or he, I'm sorry, they shared a link to ChatGPT post where they asked to calculate this, and I've looked at that. So,
right here.
And that was another super chat. So, hey, I'm helping you out, Donnie, by like making sure we bring up all your super chats.
Appreciate it.
And again, I would also agree that your commentators and the questioners been. Well, I haven't looked at all the comments. I don't know how respectful they are. I'm sure not all of them are terribly respectful, but many of them are. But the questions have been very good, and I appreciate that. So, but to go back to the question that they were just asking, that's exactly perfectly fine, right? Like the bigger the shape space that's available for evolution to occur, like if it's huge, that just means that there's that much random mutation that can result in novelty. You don't have to explore the whole space. There's a lot of work on this. This is again kind of getting outside of my area of expertise. But just the fact that there's a big space to explore doesn't mean that that's some sort of hindrance or restriction for how wandering random mutation and then selection via natural selection or other evolutionary mechanisms can result in change that actually is beneficial. That actually is a benefit. And so this question also applies to what they were asking about for all the variety of small molecules. That what I understand is that number is just simply, this is the possible shape space. There are that many organic molecules that are possible, but that doesn't mean that all of those exist. Obviously not all of those exist. But because that number is so huge, that means that just with random mutation and natural selection, that there's that much to explore for different possibilities for novelty to emerge and for new possibilities to come about. So honestly, I really appreciate these questions because both of them actually really support my point of view.
Thank you very much, Dr. Thompson. Dr. Bergman, floor is yours. Go ahead.
Yeah, I would say the problem you have with evolution is that you got thousands of these molecular shapes, and they all have to fit together, work together, function together, etc. And if one or two, the shape is changed significantly, or even in minor ways, the whole thing doesn't work and it fails. And so you're talking about a complex structure. Molecular shapes are very complex, although we've been able to draw some of them pretty well now with our machines. But molecular shapes have to have a certain function, certain shape, certain set of responses, polarity and so on responses, which makes them assemble, and the shape is formed as a result of the set of attractions, and then they have to function with other molecules as well. So we're talking about the likelihood of this occurring by chance, randomly, and so on is just zero, because we're talking about an enormous number of sequences which have to produce proteins, which have to be folded properly. If they're not folded properly, if they can't be repaired, we end up then with a problem. And so this is a major problem evolution has had, which I don't think that they really have been able to solve or even attempted to solve, because they realize how difficult problem it is. This is, of course, important when you're manufacturing drugs, because your drugs basically are looking at the shape of a molecule which responds in the correct way in order to produce the response that you want in the organism. And so it's a good question, and I think we need to look in more detail at this as well, because it is a major problem for evolution. I think it's a lethal problem for evolution.
Thank you very much, Jerry, for that response. Chris, the question was for you, therefore you get the last word. Go ahead.
Sure. So I totally disagree that it's lethal. In fact, as I said, like, the fact that the space is so big just means that random mutation can find other nodes of beneficial advantageousness that can confer a benefit to the organism. The other thing is that evolution has all kinds of explanations for how mutations, even though they may be somewhat deleterious, if it happens to be deleterious, that it doesn't necessarily result in disaster for the organism. This is the concept of canalization. So there's a ton of work that's done on this. So I encourage people, this may be a new term for some of the creationists out here, but there's a lot of work on canalization that via development that, when we look at genetic systems, they're not these linear systems. Like, a lot of times when they're just drawn out as this thing does this thing, and then this does this thing, and this does this thing. I know, Jerry, in your debate with Dan Cardinell, that you showed webs of genes and how they interact with each other in the interactome. And that, as you were saying, that it's actually a lot more complex than just even what you were showing. And that's because most of what happens within the cell and most of these gene products are actually incredibly sticky, that there's all kinds of Rube Goldbergian ways of just things being sort of, going up and down and not actually functioning like a well-oiled machine. And instead that it's like just this mishmash of stuff that somehow has some sort of function. And when you tweak something a little bit, that other redundancies that are within the system sort of just support that, the canalization of the feature to actually survive. So we actually have really good explanations for that. And the fact that the feature space is so huge means that, we're not limited. We're not limited in much at all. Like, that there's actually just tons of room. And I saw that Alyosha pointed out they, them is their preferred pronoun. So I really appreciate their comment for this.
Okay. Dr. Thompson, thank you very much for that final response on our final question for tonight's Great Origin of Species debate. Again, I want to say I appreciate the audience Q&A, the audience interaction, the comments, the questions, the challenges, and of course the super chats. I really enjoyed this. And this is what I was looking for, a good comprehensive exchange on the topic. Gentlemen, we have closing statements. 5 minutes each to wrap up our night here together. Good opportunity to wrap up our thoughts, wrap up our points, and address anything we feel has been left hanging in light of everything brought up and discussed. Dr. Chris Thompson, we are going to give you the floor for the first closing statement. And so please, whenever you're ready, just let me know. And again, you got 5 minutes. Go ahead.
Okay. Yeah. And I don't know if I'll necessarily take the full 5 minutes, but once again, I want to appreciate the opportunity for being here. Definitely a spirited debate with Jerry. I think that it's a pity that Richard Dawkins ran away from you a long time ago, because like you're a nice guy, you're actually well spoken, and you're a spirited debater, and you bring up a lot of good points. So he missed out on a good time. I want to say that I think that I made a good case for the exactly that natural selection, random mutation can confer the ability for speciation to occur, and that I think that creationists would have to agree with that, at least within kinds. Hopefully, that I've given this audience a lot to think about. That we need to look for where we actually agree on things, and that if there are phylogenetic trees that that are based on molecular evidence within a kind, or from my case, I would say within a family, that there has to be some explanation for the novelty that we can see within that kind, right? Why is the main wolf look the way that it looks relative to the bush dog, despite the fact that they are more similar to each other than they are to any other canid? I think natural selection, random mutation, and other evolutionary mechanisms, genetic drift, neutral mutation, these kinds of things are excellent ways of explaining that novelty because it works. I don't think that we need to consider magical intervention for that as well, right? So, because if you do, that does not work with the evidence as it is. I clearly made that that's the case when you're talking about within a kind. So on the origin of species, right? Speciation is something that occurs, and I think that it's something that the young Earth creationist community really needs to face, and that getting bogged down in debates about, is the chimpanzee Y chromosome different from the bonobo Y chromosome, and how is that different from the human Y chromosome or the orangutan Y chromosome? You're harking on the little details that I get why it's important to you, because the thing that you care about most is humanity, and that it's a very special creation that obviously has a special relationship with your Lord and Savior Jesus Christ, and the Holy Ghost and God, the three of them. But the other thing, but you can't ignore everything else. You have to look at the entire picture, and you have to be able to take into account the entire picture. Why within these 6 families of the muroids do we see, on the order of around 2,000 described species that are already described? There's probably more than just those, and all the variation that we can see within each individual family. Are these individual created kinds that there was only a single male and a single female that came off of the ark, and somehow you've got so much genetic variability and so much variation that you can result in a wide range of different species, 900 different species within just one kind? And you need to be able to take that into account. You have to have some sort of explanation for that, but you don't. You cannot get that level of speciation to occur that fast. And of course, that's true for every kind. You can even look at the smaller kinds. I mean, like, this is something I talked about with T-Rock at one point, the bears, right? We've got only 8 species of bears, but if you look at the phylogenetic evidence, and if you look at the molecular dating, it does not line up with archaeological evidence. Like, polar bears would have to have evolved in the 1700s in order to fit with the molecular dating, if you assume a creationist model. And it doesn't work, because we know polar bears existed before the 1700s. We have archaeological evidence going back thousands of years of that polar bears were there. But based on the genetic similarity and the differences between the brown bear, the black bear, spectacled bear, it would have to have evolved and speciated within the last 300 years. And it does not make sense. It simply doesn't work. So I like this debate on the origin of species. I think that it's an important debate for the creationist and evolution debate. I think it's also just a fascinating topic from an evolutionary perspective. So that's one reason why I'm well studied on it. But it's something that that I think the creationist community really needs to take seriously. And mostly it just gets ignored, which is a pity because there's a lot. I mean, there's a lot of just interesting issues. Like if your model's real, it has to be able to explain the pattern that we see in life, and it doesn't at this point. Maybe you can come up with another model to make it work, hopefully. So I'm going to leave it there. Thanks again. I really appreciate the debate. Again, I appreciate Jerry for being so kind and being a spirited debater, and Donny for giving me this opportunity.
My pleasure, Dr. Chris Thompson. Appreciate that closing statement, that 5-minute closing statement. Dr. Jerry Bergman, I will now reset the timer and hand it over to you for your 5-minute concluding statement. Go ahead.
Okay, my main focus is how do we get from molecules to mankind? And that's basically what evolution is. And I tend to ignore the details. I look at the overall picture. The source of evolution is random mutations and natural selection. And it's clear to me, especially in the medical area, with random mutations and natural selection, you will never get life. You will never get from the molecules to man. It's not going to happen. We know too much about mutations. Mutations are very well studied in the area that I was in, cancer research. We know a great deal about mutations, and we know there's no way that mutations can, as a whole, improve the organism. You have a few mutations which can be beneficial, can be helpful. That's not the issue. The issue is, can we start out with, period, molecules, and can we get, using mutations and natural selection, can we ever end up with human beings or the animal world and the variety that we see in the animal world? I agree there's issues relative to how in the world do we end up with this variety in the animal world. That's an issue which both creationists and evolutionists have to try to grapple with. But I'm saying that the core of evolution, it will not work. And again, my experience in 3 degrees in the medical school, we learned a lot about the body. And one thing was very clear, was that it is very well designed, it works very well, and there's no way that random mutations can produce organisms as humans. It's too well designed, although, and that's true, the word design, the word design was ignored. But on the other hand, it's clear from my courses, especially when you look at the structures like the eye in detail, you can see that it's very well designed, very well put out, and very well systemically connected with the rest of the body. And so, in fact, I know medical doctors, people in the medical area, are more apt to accept creationism, and engineers, because they see design that's there in the world. And I think all too often evolutionists don't see the design because they're not looking for it. They're looking for mechanisms relative to evolution that explain the world. And to me, they've got to take their glasses off and have to look at the world the way it is. And it's clearly extremely well designed, ingeniously designed is the best way to put it. And that is very clear from my experience teaching students and students' response to the well design of the body and of life. And so that's my main focus. You'll never go from random mutations, natural selection. It will never produce the world of life that we see. It cannot happen. And that's very clear from the evidence. And so that's my brief summary.
Dr. Jerry Bergman, thank you very much for that concluding statement. Gentlemen, again, I really appreciate the stimulating discussion tonight. And you both were very professional, gave me an easy job tonight in terms of moderating. And so again, thank you for your time. To the audience, I appreciate all of the interaction, the engagement, the questions, and of course, the super chats and comments. And so, gentlemen, we're going to wrap things up for tonight here. This was the Great Origin of Species debate. I hope you had as much fun as I did. And until we meet again, Standing for Truth is out.